Hello, everybody, and welcome to day 2 of FPWR's Research Symposium. We are thrilled to have you all here again, joining us for our second day. Hopefully you had an opportunity this morning to maybe check out the posters. There are some very exciting poster topics. We had a huge submission of abstracts, and we're not able to accommodate everything in the talks. And so we hope that you'll take an opportunity to look at those. There are also pre-recorded poster walkthroughs on a YouTube channel that you can look at as well. We, of course, want to start off by thanking our amazing sponsors listed at the top of the page, including Soleno Therapeutics, Levo, Radius, Harmony, Inversago, Saniona, Consonance, and Novo Nordisk. We are incredibly grateful and thankful for their support in helping us put on this research symposium for all of you. We're going to get started this morning. We have some exciting talks, a lot of talks in the area of neuroscience and neurobiology, and then transitioning into some behavior and additional work in the Magil-2 model. So we are going to start off with Kenichi Yamada speaking with us about the altered functional network architecture of the brain in Prader-Willi syndrome.
Hello, I'm Kenichi Yamada from Japan. I have worked as a physician scientist in the field of pediatric neurology and developmental and behavioral pediatrics. One of my research focuses to elucidate the mechanism of the brain-behavior relationship in Prader-Willi syndrome. Today, I would like to share my recent report on characteristics of functional connection within the brain in individuals with PWS based on my research project of 20 years. Neuroimaging investigations have non-invasively revealed some brain characteristics of individuals with PWS. The results have indicated that not only hormones, but the brain also contributes to the pathophysiology of behavioral repertoires observed in PWS. Conventional magnetic resonance imaging (MRI) studies have detected the structural alteration of the brain at a macroscopic level, such as pituitary structure with altered signal intensity, cerebellum specifically in posterior region, and others. Moreover, alterations in white matter microstructural integrity were detected by using diffusion tensor imaging. The results were consistent with clinically relevant brain regions in PWS as a clue to understand structural connection within the brain. Since the 1990s, functional MR imaging has enabled real-time functional analysis in sub-minute to second order. Classically, a specific condition called a task was set and the participant was required to do some action during the serial scans to detect statistical differences of brain image contrast between the task on and the task off. Resting state fMRI, an advanced form of fMRI, needs no action during the scan. This method calculates correlation of time course and image signal intensity within the whole brain simultaneously under the resting state. The pioneering resting state fMRI studies in PWS have shown correlation with regions indicating altered functional connectivity. I call this as FC. Using seed-based connection analysis, SCA, these studies have identified brain structures, including the prefrontal and anterior cingulate cortices, limbic structure, and basal ganglia. From a clinical standpoint, however, if we consider the difficulties associated with detecting both enhanced and attenuated connectivity linked with clinical behavior characteristics, a better understanding of the properties of the FC framework might be more beneficial. Recent further development introduced the combined use of independent component analysis, ICA, and SCA, which has enabled the analysis of such connectivity properties in the brain with neurodevelopmental disorder. Here, we aimed at detecting brain FC characteristics in individuals with PWS by using resting state fMRI analysis. We specifically focused on the attenuated and enhanced FC associated with behavior by using ICA and SCA. An age and sex-matched case control study in a single institute was designed. All participants with PWS were recruited through advertisements and support groups for PWS. The control participants with typical development, TD, were recruited from a local community around our city. As a result, 12 individuals with PWS and 14 age and sex-matched healthy controls with TD participated in the study. Comorbid psychiatric disorders were identified according to the DSM-5. The behavioral characteristics were assessed by using the Autism Spectral Quotient, AQ. No individuals had a history of any other neurological diseases or traumatic brain injury that resulted in any neurological sequelae. By the way, don't you like having an MRI scan which act with loud noise? Boom, 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 beep, 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 beep. Nevertheless, even the slightest head movement during the scan can lead to image degradation and fMRI analysis. First, to become familiar with the imaging procedure, and as a result, to reduce imaging artifact, the Zero Tesla mock scanner preparation system that we have developed in-house was utilized as a rehearsal session before the actual imaging. Next, a checklist was utilized to assess whether the participants' understanding and mental rehearsal fulfilled the criteria. As a result, our preparation strategy consisted of 4 steps as shown in the figure. Audiovisual aids were utilized to maximize comfort and effectively reduce anxiety in the real MRI scanner. No sedative agent was administered, nor was it required. The detail was shown in my paper. All imaging studies were performed by using a GE Signa Lilex 3.0 Tesla imaging system with an 8-channel phase array reticle. 15 axial slices that cover the whole cerebrum were obtained with single-shot gradient echoplanar pulse sequences. Image scanning was performed in non-wake state with participants viewing a clear screen with a fixation point via mirror reflection. Head motion of the participants during the scan was estimated from acquired data by using an in-house script based on translation and rotation of rigid body assumption to ensure that the range of translation and rotation could be limited within plus minus 0.6 millimeter in all studies. The scan time for each fMRI data acquisition was about 10 minutes. All fMRI data processing was performed by using the CON toolbox within SPM12 based on MATLAB. The image processing in detail was described and shown in my recent paper. The seed region in SCA were defined in 106 areas by using the default setting. To eliminate false positive results associated with image distortion artifacts, the regions used as seed regions in cerebellar structures were excluded from the current study. The voxel-based comparison between the two groups was performed by using ANCOVA with age and sex as covariates. Cluster threshold was set at p-value less than 0.05 with family-wise error corrected. Cluster size was false discovery rate corrected, and voxel threshold was p-value less than 0.001 without correction. The main results are graphically shown. 19 independent components showing significant differences in FC between the PWS and the TD control groups were detected and adopted. The components were scattered in the frontal, parietal, temporal, and occipital lobes of cerebral structure. The structures with the most spatially extensive components were the superior lateral occipital complex, followed by right postcentral gyrus. The most statistically significant components were the superior lateral occipital complex, left angular gyrus, left superior parietal lobe, and posterior supramarginal gyrus. Correlation analysis showed that autism questionnaire scores were found to be significantly correlated with FC in the right inferior frontal area. The PWS group exhibited differences in FCs over the several brain regions as indexed by clusters compared to that in the TD control group. Each seed region is shown as a gray-colored ball. The representative correlation clusters detected were as follows: front parietal, supramarginal gyrus, and superior parietal lobules, temporal parietal, front temporal parietal, and hippocampus and brainstem structures, along with interhemispheric FCs in each connection. Altered FCs detected indicates bidirectional contributions, that is, enhanced and attenuated brain circuitories in the front polar parietal versus the dorsolateral prefrontal parietal connections. Both brain regions have been considered as important in PWS. The functional alteration in our study is consistent with previous research and provides a promising framework for understanding the difficulty in flexible social behavior associated with PWS. Taken together, our findings indicate that circuit-based functional alterations indeed exist and involve front parietal connections. Although the reward and inhibitory circuitories are supported by the frontal striate parietal circuit, impulse control difficulties have been associated with the front polar parietal temporal network, which has also been implicated in PWS. The significant correlation between FC at the specific brain region and the autistic features as indexed by the AQ ensures functional contribution. The right inferior frontal area detected has shown to be associated with inhibition, attentional control, and task switch. Considering that there has been debate But the similarity and the difference between ASD-like traits in PWS and typical autistic traits, our findings might provide a clue to help decipher a constellation of ASD-like symptoms in PWS. Further evaluation with a larger sample size comprising the genetic subtypes is highly warranted. In conclusion, altered functional connectivity indeed exists in the representative brain regions of the brain in individuals' PWS. Enhanced versus attenuated connectivity in the specific circuits are consistent with the behavioral features in PWS. The finding may contribute to not only behavioral phenotype in PWS, but also other behavioral characteristics in neurodevelopmental disorder. Piotr thanked that Prader-Willi Syndrome Association Japan, the Takenoko no Kai, and Emiko Aiba for supporting neurodevelopmental assessments in each participant. We also thank and pay tribute to my boss, Stondo Nakada, Brain Research Institute, University of Niigata. Finally, I would like to show my appreciation to the participants with PWS, their families, and professional caregivers who shared their knowledge about the behavioral characteristics. Thanks so much for your attention.
Thank you so much for providing us with that talk. Dr. Yamada, we're having some technical difficulties getting him hooked up for the live Q&A session. But hopefully we'll be able to resolve that, and possibly there are some questions that have come into chat, and we'll be able to answer those or reach out to him directly. We're going to move on to our next talk from Caitlin Victor, who's going to be speaking with us on the topic of genotype to phenotype and how the molecular changes in PWS neurons may relate to risk and susceptibility of autism in PWS. Thank you, Caitlin.
My name is Caitlin Victor. I'm a graduate student in the lab of Dr. Larry Ryder at the University of Tennessee Health Science Center in Memphis. And today I'm going to be talking to you about the molecular changes in Prader-Willi syndrome neurons, revealing clues about increased autism susceptibility. So the model system that we work in our lab is dental pulp stem cells, and these are neural crest-derived stem cells that reside inside of the tooth cavity, and they are depicted here in this picture down here by these yellow circles within the pulp cavity. These are multipotent stem cells. They've been differentiated into a variety of cell types, including neurons and glia, adipocytes, and many other cell types. And what's so great about this system is that these stem cells are easy to collect. So children naturally shed their baby teeth. There's no need for any invasive procedures to obtain these stem cells. And we're able to collect samples from long distances using this method, which is really important when you're studying rare neurogenetic syndromes. So how we collect these is an interested parent emails our lab a genetics report so we can confirm the diagnosis. Then we send them a tooth kit, which includes a tube of media, a consent form, and instructions. Once the tooth is lost, it's sent back to us where we crack it open in the lab and we mince it both mechanically and enzymatically, and then we plate it with growth media. And typically we'll start seeing the stem cells forming after about 5 days, but it can take up to a month. And using this method, we've been able to collect 200 individual cell lines across 14 different syndromes, with the largest group coming from the Prader-Willi community, which I've highlighted here. We have 43 individual Prader-Willi stem cell lines in our collection. And what we do with our stem cells is we differentiate these into neuronal cultures. And this involves a 3-step differentiation protocol. So the first step is epigenetic reprogramming, which erases the epigenetic marks on the stem cells and makes them more flexible to become what we need them to become. The second step is a neural induction phase, which includes several neurotrophic factors and different PKC, cyclic AMP activators, followed by a 4-week neuronal maturation. And over here in this picture, you can see that the stem cells have a flat fibroblast-like morphology prior to differentiation. And then post-differentiation, they develop this pyramidal morphology with neuronal projections. Over here, we have a Western blot showing that these stem cells are immunonegative for common neuronal markers like GABA-A receptors and MAP2. But post-differentiation, they are positive for these markers. So this is a picture of the 15th chromosome, and the Prader-Willi critical region is displayed here by this yellow square. And within this region, there are genes that are specifically expressed from the paternal allele, and these include MAGEL2, SNRPN, and SNRF. And so if you inherit a paternal copy of this allele with this region deleted, you're part of the deletion subtype. If you inherit 2 maternal copies of this allele, which means you're still not getting expression of these paternally expressed genes, that is part of the UPD subtype. And within the UPD subtype, they tend to have milder PWS phenotypes, but there is an increased autism risk. And in our lab, we're interested in understanding the differences between these subtypes, as well as the difference between Prader-Willi neurons versus neurotypical control neurons. So to look at these differences between these subtypes versus neurotypical control, we performed RNA-seq using our dental pulp stem cell-derived neurons. We used 4 individuals per group, and these groups include neurotypical control, Prader-Willi deletion, Prader-Willi UPD plus autism, and minus autism, and I'll get into that in a few slides. This Venn diagram here represents all of the significantly differentially expressed transcripts versus control for each of the subtypes. And I want to draw your attention to these 12 in the center. So these are common to all of the Prader-Willi subtypes, regardless of specific genotype. And within these 12, we found things that you would expect to be there because they're within the Prader-Willi critical region on the 15th chromosome. And these include MAGEL2, SNRPN, and SNRF, but we also found genes outside of that region that may be contributing to a Prader-Willi phenotype. So we followed up on several of these that met our RNA-seq cutoff, which was at least 10 counts or more. And I just want to highlight a few of the interesting things that we found within these genes. So PEX10 is a peroxisome biogenesis factor, and mutations in this gene lead to a syndrome called Zellweger syndrome, who has primary phenotypes that are similar to Prader-Willi, such as hypotonia and neurodevelopmental delay. Also, SNTB2 and ATP7A are both involved in the secretory granule pathway. And we know that this pathway is disrupted within Prader-Willi via likely the MAGEL2 mechanism. And each of these is associated with different syndromes. So SNTB2 is associated with muscular dystrophy, and ATP7A is associated with a disease called Menkes syndrome. And the primary phenotypes with this syndrome are hypotonia, hypothermia, and failure to thrive. So within the Prader-Willi UPD class, the rate of autism is much higher than in the deletion class. So it's reported that about 35% of UPD subjects will meet the criteria for autism versus 18% of deletion patients. And how we assess possible autism in our subjects, since it's not a clinical diagnosis, is we include a social communication questionnaire with each of our tooth kits. And this is a validated parent questionnaire that's used to assess possible ASD, or the likelihood that a subject has autism, with a score of 15 or above indicating possible ASD. So down here we have each of our groups, control, deletion, and Prader-Willi UPD, stratified by SCQ score. And as you can see in the UPD class, you have a group that clusters below 15, meaning within normal limits. And then you have a smaller subset that groups above the cutoff of 15, around 20, which indicates possible ASD. And so of the 18 patients we have here, 6 of them meet the requirements for possible ASD, which is 30% of our sample, which matches pretty well with the 35% reported in the literature. So again, to look at what is specifically different between these UPD cases with autism versus those without, we plotted another Venn diagram, except in this one, each of these circles represents the significantly differentially expressed transcripts versus the UPD plus autism group. And here I want to draw your attention to these 312 that were specific to the UPD class plus autism versus minus autism. So we performed a David enrichment analysis on these 312 transcripts, and that helps to identify enriched Go terms within your data set. And what we found when we did this David enrichment analysis was that all of our enrichments were in mitochondrial function, components, and different mitochondrial areas, including the mitochondrial inner membrane, the electron transport chain, the mitochondrion, and additionally hydrogen ion transmembrane transport, which is important because hydrogen ions drive the electron transport chain. So we took the identified transcripts in our David analysis, and we plotted a heat map across each of our 4 groups here. So we have control, deletion, the UPD class minus autism, and the UPD class plus autism here at the end. And as you can see, for the majority of these transcripts, they're significantly reduced in our plus ASD group. So this was our first indicator that maybe something's going on with the mitochondria within this subset that is leaning them towards an autism diagnosis. And when you look in the literature, mitochondrial dysfunction has previously been associated with autism. In one study, they found that throughout postmortem ASD brain, there was reduced mitochondrial electron transport chain protein expression. Additionally, in the literature, between 5 and 80% of ASD subjects also present with mitochondrial dysfunction. And although that's a very wide range, at either end of the spectrum, it's greatly increased from the 0.01% of the general population that presents with mitochondrial dysfunction. And mitochondria have a very important role in neurons, so they help maintain ionic gradients and calcium homeostasis at the synapse. So any dysregulation of neuronal mitochondria could disrupt these gradients and thus impact neurotransmitter release. So next we wanted to look at the mitochondria within these neurons to see if there was any phenotype. And so what we did was we stained our cultures with beta-tubulin, which is just a general morphology stain. And we also used TOM20, which is a outer mitochondrial membrane stain. So it stains all the mitochondria within the neuron. And up here at the top, we have a UPD minus autism neuron. And as you can see, by the TOM20 staining, the mitochondria are nicely dispersed throughout the cell body and out into the projections, versus our plus ASD neuron here at the bottom, where you start to see this aggregate phenotype, and you're seeing these mitochondrial aggregates within the cell body and also out into the projections. So I needed to create an assay to measure this phenotype because we actually want to measure dispersion as opposed to fluorescence intensity. So to do this, I used a program called Amaris, and what this program does is it creates a mask over the channel that you're looking at to give you a total area of the protein that you're looking at. So again, I used beta-tubulin just to get the total neuronal area, and that's here at the top. And I used TOM20 to get the mitochondrial area within each neuron. And over here on the left side, we have a neuron with the aggregated mitochondria phenotype. And from the measurements, you can see that the neuronal mitochondrial coverage is about 22% in this neuron versus a neuron that has dispersed mitochondria where the measurement's about 44%. So over the past year, I have been performing a blinded, unbiased analysis across each of our groups with 6 individual cell lines per group, measuring at least 15 neurons per individual to determine if the mitochondria within the ASD group was significantly decreased versus our other groups. In this graph, you can see that here at the end, the UPD plus ASD group shows a significant reduction in neuronal mitochondrial coverage versus all of the other groups. So the conclusions from this project were that we identified 6 differentially expressed transcripts that could be related to a common PWS phenotype in our DPSC neurons. We found a significant reduction for downregulated mitochondrial transcripts in the plus ASD versus UPD minus ASD groups. We observed differences in the mitochondrial localization of the UPD plus ASD group. And additionally, I was able to create an assay to measure this mitochondrial aggregate phenotype, and we were able to confirm that the mitochondrial volume within the UPD plus ASD neurons is significantly reduced versus the other groups. So to expand on these studies, the next thing that we want to do is assess the mitochondrial bioenergetic function within these neurons. And to do that, we plan to use a seahorse analyzer, which uses oxygen consumption rate as an output to identify defects in specific components of the electron transport chain. And the goal with this study would be to determine if it's just that the ASD neurons have less mitochondria or whether these mitochondria are actually defective. Additionally, in our RNA-seq data, we found this transcript, PPARGC1A, is significantly reduced in our UPD plus autism neurons. And this transcript encodes a protein that is a master mitochondrial biogenesis regulator. And actually, activation of PGC1 alpha has been used as a therapeutic compound in mitochondrial disorders. So what we'd like to do is treat our neurons with a PGC1 agonist and see if we can recover some of the phenotype that we found. Additionally, we want to use other small molecules that have shown promise in clinical trials and in vitro to be mitochondrial enhancing, like CoQ10, folic acid, creatine, and L-carnitine to see if we can rescue this phenotype. And finally, we'd like to use genetic manipulation of UB3A to see if this has any effect on the mitochondria within these ASD neurons. So within the UPD class of Prader-Willi, you have an increased, or you should have increased levels of UB3A, as this is expressed from the maternal allele. And increased levels of UB3A has been associated with autism risk, and UB3A has also been shown to modulate mitochondrial function. So the goal here would be to attempt to reduce UB3A expression in the Prader-Willi UPD neurons using an siRNA, and then perform mitochondrial analysis to see if we're able to recover any of the phenotype that we have discovered here. So with that, I would like to thank the Foundation for Prader-Willi Research and the Prader-Willi community and families for their continued support of our work, the University of Tennessee Neuroscience Institute, all of our collaborators, and of course my mentor, Dr. Ryder, and the other members of the Ryder Lab. And with that, I will take any questions that you may have.
Thank you, Caitlin. Great talk. We have a lot of questions that have come in, so your talk has spurred some interesting comments. The first one is from Hui Yu, who wants to know whether or not you've compared your RNA-seq results with any post-mortem tissue. Specifically, there's a publication by the Faruqi group looking at post-mortem hypothalamic RNA sequences and whether or not your genes correlate with what they found in that publication.
I have not specifically looked at that paper. I will actually do that this afternoon, though, because I'm sure that would be interesting to see if there's any overlap there. I'm certainly always interested to check the overlap with our results, with anything that's relevant, and specifically looking at the hypothalamic region would be really interesting.
There's a couple other questions on what other sort of subgroups you have looked at or may want to look at in the future. Have you looked at ASD only? And the other question is, have you looked at the CIS population, the Xiaopyang population at all?
Yeah, so I actually did just generally look at the mitochondria in the Xiaopyang neurons, and I didn't see this specific clumping phenotype. That's not to say that they don't have a reduced volume, mitochondrial volume, as I didn't do an extensive analysis. I kind of just did a quick general check to see if we saw that clumping phenotype, which I did not, but that doesn't mean there isn't a mitochondrial phenotype, and we do plan to investigate more syndromes and specifically Xiaopyang to see if there is a mitochondrial phenotype associated with that group, because they do have higher rates of ASD. And then was there another question? Did we look at?
We have a couple.
Okay.
Oh yeah, sorry. Did you look at ASD only? Was what Dr. Forrester was asking, Dennis Forrester.
We looked at the deletion cases. I think is the question maybe that did we look just at the ASD cases in our group? And we did the general RNA-seq across different Prader-Willi subtypes, including deletion and the UPD cases without autism. And there did seem to be a little bit of a mitochondrial phenotype, but not as severe as in the ASD group. And there was not that specific clumping in the deletion subtype.
Clementina was wondering if the mitochondria are damaged and are perhaps being targeted to lysosomes.
That would be
If you
interesting
had
to
the
look
opportunity to
at.
check that.
Yeah, that would be interesting to see. We could do a co-stain and see if there's some lysosomal targeting going on. And we do want to check the mitochondria health in general, because what we've seen is that there's a phenotype, but we're not sure yet if those mitochondria are actually damaged or if they are working correctly. So that's something else we plan to do in the future.
Okay, let's see. Teresa Strong is asking whether or not low PGC1A / decreased mitochondrial volume has been described in general non-PWS autism.
Yeah, so I have not seen PGC1 alpha specifically associated with autism, but certainly decreased mitochondrial volume. Specifically, I read a paper where they used the dental pulp stem cell neurons to study Fragile X, and that's kind of where I got a lot of my ideas for this project. And they saw kind of not the exact phenotype we're seeing, but they measured mitochondria in the neuronal projections, and they saw a decreased amount of mitochondria. So certainly decreased mitochondrial volume has been associated with ASD, and PGC1 alpha contributes to this. So I haven't seen it specifically associated, but I would imagine that there is a component there where it is associated with that biogenesis gene.
Okay. And then sort of along those same lines, Jim Resnick was asking if we know if the frequency of autism is increased in mitochondrial disorders in general. I mean, obviously there's a lot of different mitochondrial disorders, but what the autism rate is.
Yeah, it seems that like within the general population, mitochondrial disorders affect about 0.01% of us. And in children with autism, they tend to have a much greater frequency of mitochondrial disorders. So I would say that yes. And when you read different reviews, the range is quite vast, but certainly there is an increased risk of autism in mitochondrial disorders, I would say, from the literature that I've seen.
Okay. And then lastly, a follow-up question from Clementina was asking if you're seeing increased oxidative stress.
So I haven't checked that yet. Findings. Yeah, so I certainly think that is something to look at, reactive oxygen species. If there is increased oxidative stress, not only that, but how these mitochondria are able to respond to oxidative stress is something we'd like to look at. There's so many avenues of mitochondrial function that we'd like to test, and I think that oxidative stress is certainly an important one that we should check.
Great. Thank you so much.
Thank you.
If you have additional questions, please go ahead and put them in the chat. We can send those along to Caitlin. As a reminder, if during any of the talks you have any questions or comments, please put those ahead in the chat so that we can prompt great discussion. We're going to move forward into bringing into more of a mouse model. Dr. Isles is going to talk with us about a comparison of mouse models to reveal a molecular distinction between psychotic illness in PWS and schizophrenia.
Okay, so I'm Anthony Isles from the University of Cardiff, and I'm going to talk about some work that we've been doing over the past 15 years now, looking at mouse models and trying to model the psychotic illness and look at the biological basis of psychotic illness in Prader-Willi syndrome. So, as I'm sure you're all aware, in addition to the core diagnostic characteristics of Prader-Willi syndrome, individuals can sometimes suffer from a number of other problems, including behavioral and psychiatric problems, such as obsessive-compulsive disorder and negative affect and psychotic illness. And when I first started working on Prader-Willi syndrome, it was with Tony Holland's group, and they had recently published some work showing that these negative affect and psychotic illness were more prevalent in certain Prader-Willi genotypes. So this is a schematic showing the Prader-Willi locus that I'm sure you're all very familiar with. So we have a number of paternally expressed genes and 2 maternally expressed genes, UB3A in particular being important here. And of course, the loss of paternal gene expression is what is causal in Prader-Willi syndrome. But what Tony and his colleagues have found was that these 2 genotypes here, maternal uniparental disomy and paternal imprinting center deletions, have a far greater prevalence of psychotic illness. That's to say that it's not the case that psychotic illness never occurs in paternal deletions, but that these 2 genotypes have a higher prevalence for around 50 to 60%. And the common factor for these 2 genotypes, as well as a loss of paternal gene expression is that they have overdosage of the maternal genes, in particular UB3A. And this work has been replicated by a number of others over the last few years. So we wanted to model this in animals, and of course, as I'm sure, again, you're well aware that the animal locus on chromosome 7 is quite homologous and there's a high degree of synteny between humans and mouse. And so we decided to take the approach of looking at two different mouse models, one of which we thought would be more prone to the phenotypes of relevance to psychotic illness. So the first animal that we looked at is the minimal critical deletion model. And this has a deletion, paternally derived deletion, that spans the SNO RNA SNORD116 and also deletes IPW. And this replicates the minimum deletion that is seen in rare cases of individuals who have the core phenotypes of Prader-Willi syndrome. The other model that we used was the IC deletion, the imprinting center deletion. And this is one of the genotypes that is associated with the increased psychotic illness in Prader-Willi syndrome. And this mouse model, as well as having loss of all paternal gene expression, we and others before us have shown that it also has overexpression of UBE3A. So we're comparing these two models to look for differences in terms of types of relevance to psychotic illness. But one thing I want to emphasize is that actually phenotypically both have very similar phenotypes in terms of growth. They're both growth retarded, and they also both suffer from early life failure to thrive. They also both have endocrine problems and changes in ghrelin levels, and they also both have incidences of hyperphagia. So there is a lot of commonalities, but we're looking at differences between these two different lines. And so the behaviors that we looked at that are particularly relevant to psychotic illness are sensory motor gating. And here we measured this by looking at two different measures: the acoustic startle response, so that is the response to a loud stimulus and the startle response of the animal, and also prepulse inhibition of the startle response. So this is where a stimulus is paid just before the onset of the startle stimulus, and that inhibits the overall startle response. And this is a measure of the animal's ability to integrate sensory information with motor function and output. In addition, we looked at a number of aspects of cognition, and I'm going to focus here on the 5-choice serial reaction time task, which you can see the animal performing here, where it has to scan this array of holes here, and a light will appear briefly in one of the holes. 0.8 of a second it lasts. The animal has to nose poke in order to be able to gain a food reward. And from this task, you get a number of different measures which can reflect different aspects, including attention and inhibitory control, such as impulsivity. And the really important thing about both these different tasks is that they are highly translatable. So measures of sensory motor gating, acoustic startle, is used in humans, and deficits in acoustic startle and prepulse inhibition are characteristic of psychotic illness in people. And similarly, the 5-choice serial reaction time task, there is an analogous version in people using a touchscreen, and this can be used to measure aspects of cognitive decline. So as I said, we did a whole range of behaviors, and I'm going to summarize these because I have talked about these in the past, and a lot of this data is published. But suffice to say that in a number of measures, both the critical interval Prader-Willi mouse model and the IC mouse model were equivalent to their wild-type littermates. So in our hands, in terms of anxiety, for instance, across two different measures, there was no difference between the critical interval and their wild types, or the Prader-Willi ICs and their wild-type littermates. But in those two measures that I've just highlighted, sensory motor gating and attention as measured in the 5-choice serial reaction time task, what we found was that generally the critical interval mice were equivalent to their wild-type littermates. And the only difference we found was a decrease in the startle response of the critical interval mice relative to their wild types. In contrast, the IC mouse model showed a number of deficits across these different measures. So they showed an increased acoustic startle response and a decreased prepulse inhibition of startle response. And that is a characteristic pattern of behavior that's seen in individuals with psychotic illness and in animal models of schizophrenia and other psychotic illnesses. In addition, we found deficits in attention in the 5-choice serial reaction time task. Specifically, we saw decreases in accuracy of responding, an increase in the rate of omissions, and a slower reaction time. So these are all indicative of attention. So we're quite, we think that we have a mouse model that's showing endophenotypes of relevance to psychotic illness. So the next question that we wanted to ask was, are there brain gene expression changes that may reflect these behavioral differences between these two mouse models? So to do this, we performed RNA-seq of whole neonatal brain, and we did two comparisons. We did the critical interval mouse model to its wild-type littermates and the IC mouse model to its wild-type littermates, this matching the behavioral data that I've just presented. And by doing it this way, we could generate two different pools of gene expression changes in the different animals and identify common changes that may be PWS general changes that are related to the core phenotypes, perhaps, and then separately distinct changes. And in particular, distinct changes in the Prader-Willi IC model might be of relevance to the cognition and also possibly relevant to psychosis. So here is the gene expression data from both these animals, and what we can see is that there are some changes in the critical interval mouse model, but there's a far greater number of changes in the ICs. Importantly, the Prader-Willi genes that have been disrupted in both these models show significant decreases in expression, but in the ICs you also see a number of changes in genes that might be of relevance to brain function, such as PER1 here, which is a circadian rhythm gene, and also PLP1, which is a gene that's been associated with psychiatric illness and neurodevelopmental disorders. Our read depth from our RNA-seq analysis was large enough to also allow analysis of isoform usage. So this is where genes may not have changes in their overall levels of expression, but may have changes in the different isoforms that are used and the proportion of those isoforms. So the proportion of alternate splice forms of those genes. And again, we saw a number of changes in the critical interval mice, but far more changes of this nature in the ICs. And I just want to give you an example of a change in the ICs that might illustrate why this is important to look at. So this is the gamma-3 subunit of the GABA-A receptor. And in the ICs, we saw no change in the overall level of gene expression for this gene. But what we did see was a change in the isoforms. So here, this middle isoform of this gamma-3 receptor subunit gene is the most often used isoform in the brain. And this contains the neurochannel domains, including the neurochannel membrane domain. And you can see that it's the most increased usage here. But what you can also see is that the ICs have a significant decrease in this isoform. And in addition, they have concomitant increases in a truncated isoform, which is less functional as it lacks the neurochannel domains. So although there's no change in gene expression, what we're seeing for this particular gene is a switch in the proportion of the isoforms, which might have functional relevance to brain physiology. So I've highlighted a couple of gene changes there, but by doing so, it's there's an inherent bias in doing this, in that you're kind of driven by your prior knowledge. And what we wanted to do was really to systematically investigate these gene changes in a much more robust manner. So what we did was combine the gene expression and isoform switches to provide a list of changes for both different animals, for the ICs and for the critical intervals. And this revealed both common changes and 101 differences that are unique to the IC brain. And what we wanted to do then was explore these data collectively rather than focus on these individual changes. So the way we went about doing that was to look for enrichment of human genetic variants associated with disease. And we're looking for changes in genes in the brains of these animals that are linked to those human genetic variants. So what we're doing is taking GWAS data from studies of schizophrenia, and then separately GWAS data for studies of psychotic episodes, any and multiple, and also distressing psychotic episodes. And then as a negative control, GWAS data for chronic kidney disease. And so this GWAS data points to certain genes that may be key to these disorders. And what we're then doing is looking for whether those genes are enriched in the gene expression changes we see in our mouse models. And so what we're looking for is changes that are above chance, so significant enrichment or overrepresentation of these genetic variants in the gene expression changes we see in the mouse models. And so what you can see from this table here, this figure here, is that the critical interval mouse model, there was no enrichment in the gene expression changes for any human genetic variants associated with any of these disorders. In contrast, in the IC gene expression changes in the brain, there was a significant overrepresentation and enrichment for GWAS signals, genetic variants, that were associated with any psychotic episode and multiple psychotic episodes. So this is really nice because it confirms or validates our strategy in that we've taken behavioral data which we think is indicative of psychotic illness, and then we've followed this up in the mouse model and looked in the brains of these animals, gene expression changes that are enriched for genetic signals that are indeed associated with psychotic illness. But equally, really interestingly for me, is that GWAS signals, genetic variants associated with schizophrenia, were not enriched in the IC mouse model. And this may seem paradoxical because schizophrenia is the main widely known psychotic illness. But the way to think about this is that there is genetic liability for schizophrenia, and then separately there is genetic liability for other psychotic illnesses. And of course, there's a large degree of overlap between these two. But there are also some genetic variants that are not associated with schizophrenia and are associated with other psychotic illnesses. And it's in this proportion of this figure where those gene expression changes that we see in the IC model fall. And this was really nice because it reflects the diagnostic data seen in PWS. Indeed, when I was first started with Tony Holland, there was a PhD student there, Sarita Soni, who was doing a really nice etiological study of psychotic illness in Prader-Willi syndrome. And she repeatedly told me that individuals with PWS do not have schizophrenia. Their psychosis is different. And I guess our data provide a biological basis for this observation. And it also supports the idea that therapeutics for schizophrenia may not be appropriate for treating psychotic illness. Instead, better therapies or avenues for therapeutic development may lie in studying genes that fall in this proportion of the genetic liability for psychotic illness. So as I said, this work has been done over about 15 years now, and some of it has been published in previous papers that are listed here. And I just wanted to highlight this publication, which came out earlier this summer. And this publication ties together all these behavioral studies and also with the RNA-seq study that I've just described. And again, of course, this work involves a large number of people over the years. But again, I just wanted to highlight Simona Zahova, who's seated here, who did all the RNA-seq and did the analysis that pulled all this data together. And finally, I just want to thank my funders over the years, including the Foundation for Prader-Willi Research. And so I believe there's now time for some questions, and I'm happy to take any that we can have.
Unmute.
I was on mute. Apologies for that. We do have a couple of questions for you, Anthony. Larry Ryder was asking about your IGF-2 H19 results. He was saying these are really interesting and that they are oppositely imprinted, and was wondering if you would be able to comment on that.
Yeah, it is interesting. I haven't really thought about it in that much detail, but it is interesting because there's a lot of evidence that suggests that imprinted genes are often regulated in kind of networks. So if you perturb one or other or a couple, you often perturb others. So yeah, so the fact that you're getting changes in other imprinted genes as a consequence of the changes in the ICs, yeah, is really interesting. So yeah, it's nothing I've pursued in particular much detail, but it is really interesting.
And then Teresa Strong was asking, she's saying that there is a component of bipolar disorder in PWS, and if you've had an opportunity to compare the GWAS data for that.
No, we haven't. That would be an interesting thing to do, to look for enrichment of bipolar, because there is overlap between bipolar disorder and schizophrenia, but there is a distinction. So it might be worth looking for that as well. Yeah.
Okay. And then Dr. Ryder also had a follow-up question asking if you consider doing single-cell RNA on any of these brains, and that might provide some additional information.
Yeah, absolutely. I mean, the approach we took was kind of limited by what the tissue we had, so it was relatively crude. It was whole brain analysis, and yes, I mean, we obviously got quite interesting findings from the ICs. We got significant, 101 changes that were interesting, but we didn't get very many in the critical interval mice. And of course, other people who have done more focused studies on the hypothalamus and things like that have found a significant and larger number of changes in those animals. So I think by focusing down, doing a single cell RNA-seq would be a really interesting thing. Of course, it's just not very cheap, but yeah, it would be a great thing to do.
Teresa's asking if you have looked at or plan to look at human data sets, human tissue. I think that somewhat relates a little bit to a question that I was going to ask, which is, what would it take? What data would we need to be able to support sort of clinical care along your statement of that, psychosis and PWS is not schizophrenia, in order to be able to get that sort of more into the mainstream mental health world? Yeah,
so I was really interested in the previous speaker's talk, because I mean she was taking these iPS derived neurons and they're from these dental pulp cells and looking in, and they were different genotypes. So that's a really nice data set where it might be worth looking for overlap between the kind of things that are seen in the ICs and the things that are seen in the UPDs, for instance, in that study. So that would be a really nice comparison. And in terms of the kind of the actual translating this into the human studies, I think what we're saying is that we're providing a kind of biological basis for the diagnostic observation that's already there. So I think it's people are aware. Tony Holland's group and others are aware that the psychotic illness that's seen in PWS is not schizophrenia. This kind of just provides a biological basis. Whether it would lead to therapies, yeah, you're right. It would probably involve human tissue analysis and also focusing on some of those genes or trying drugs that affect some of those genes that have altered expression. So, for instance, the gamma subunit of the GABA A is a really interesting one, because that is very close as well to the Prader-Willi locus and sometimes gets disrupted.
Right. Well, Larry says he's ready to collaborate. Hit him up and set up a Zoom. And Elena Nikonova wanted to ask, what about non-neuronal markers?
Non-neuronal markers, as in genes that change. Yeah, there was quite a few actually. There was quite a few metabolic markers that changed that were not normally expressed in the brain and suddenly were expressed in the ICs, and they shouldn't have been. So that's interesting.
Okay. Thank you so much.
Thank you.
We are going to hear from our next speaker, Gabriela Bosco-Ortiz at Yale, who is going to speak with us about how the loss of MADRL2 alters the development of separation-induced vocalization and maternal behavior.
Hi, everyone. My name is Liz Nitlicki-Boz. I'm an assistant professor at the University at Buffalo, and I'm happy to share our work with you today on characterizing the energy balance phenotype of the MADRL2 deficient rats. I would like to thank the organizers for the opportunity to speak today. Prader-Willi syndrome, or PWS, as we know, involves numerous symptoms that are relevant to energy balance control. Individuals with PWS display overeating and weight gain, dysregulated peripheral feeding-related hormones, including ghrelin and leptin, and changes to macronutrient metabolism. Although this energy balance phenotype is well established, the neurobiological underpinnings of these changes in PWS remain unresolved. We know that energy balance is controlled by the brain, represented in this picture at the right. The brain takes information from peripheral sites, including the gastrointestinal tract, adipose tissue, and others, as well as centrally derived signals, and integrates these to exert control over feeding and weight. However, the control by the brain for energy balance is very complex, involving numerous receptors, genes, neurohormonal signals, and distributed nuclei throughout the brain. Today, I'm going to be focusing in my talk on the role of a particular gene, MADRL2, in energy balance control. So MADRL2 is one of several genes located on chromosome 15 that's disrupted in PWS. Deletion of MADRL2 plays a critical role in the energy balance phenotype of PWS. As shown on the right, MAGEL2 is expressed in the hypothalamus of mice. This is a key brain area for energy balance control, and several research groups have published data showing the importance of MAGEL2 expression in the hypothalamus for normal development and function of circuits related to energy balance. Rodent models, therefore, are particularly useful in understanding the neural underpinnings of PWS because they allow us to more fully probe neural changes that underlie altered feeding and body weight. So one rodent model that's been used in PWS research has been the MAGEL2 null mouse. And as shown in this table from a paper by Bischoff and colleagues, this mouse model does indeed display some of the phenotypes similar to PWS energy balance phenotypes, including increased adiposity. However, this model fails to recapitulate other key phenotypes of PWS, including weight gain and overeating. So body weight in wild-type controls and MAGEL2 null mice is similar, and MAGEL2 null mice actually eat a bit less in daily food consumption compared to wild-type controls. So this points to the importance of identifying rodent models with phenotypes that more closely resemble that observed for energy balance control in individuals with PWS. The recent development of a MAGEL2 deficient rat has provided a unique opportunity to further investigate the neurobiology of MAGEL2 deletion and then the key energy balance phenotypes resulting from this change. There are species differences between rats and mice in some aspects of energy balance control. And furthermore, when we think about studying the role of a particular gene in energy balance control, rats and mice may exhibit different phenotypes as a result of knocking out a specific gene. So it's possible that disruption of MAGEL2 expression in rats might produce an animal that more closely models energy balance aspects of PWS compared to the MAGEL2 null mouse. So the overarching goal of our project is to evaluate various energy balance relevant phenotypes of the MAGEL2 deficient rat. And we're using rats that are heterozygous for paternally transmitted MAGEL2 deficiency versus wild-type counterparts as controls. Our project has three major areas of investigation. The first is to assess weight gain, feeding, and meal patterns in the MAGEL2 deficient rat to determine whether MAGEL2 deficiency in these rats more reliably produces hyperphagia and increased body weight like observed in PWS. Second, we're evaluating the motivation for and preference for palatable food in this model. And third, we're measuring neuronal activation in response to a satiation signal in these rats, and looking at a few brain areas, the hypothalamus, which I mentioned, as well as the hindbrain, which is another area of the brain that serves as a critical hub that integrates energy balance information to control feeding and weight gain, but to date is relatively unexplored in PWS research. So I want to start with the disclaimer that this is still very much a work in progress. We breed these animals in-house for the studies, and we're running several waves of animals through a battery of tests to address these questions. I'll be showing you data today from the first 2 to 3 waves of animals that we've run so far and addressing these first 2 questions. So after we breed the rats, they get weaned at 3 weeks of age, and the first thing we do with our MAGEL2 deficient rats, abbreviated throughout as KD for knockdown, and then our wild type or WT counterparts, which are our controls, is to track their body weight after weaning. And we hypothesized that MAGEL2 deficient rats would exhibit increased weight gain consistent with the increased body weight observed in PWS. What we found is that the females in pink generally gain less weight than the males in blue, and that's to be expected. Male rats are generally larger than females. We did detect a significant main effect of sex here. But turning attention to genotype differences, so between wild type and knockdown animals, we detected a main effect of genotype, but actually in the opposite direction of what we had predicted. So our knockdown or MAGEL2 deficient animals tended to gain or did gain significantly less weight than did the wild type counterparts. And we found no interaction of sex and genotype. Overall, this is suggesting that MAGEL2 knockdown in rats appears to reduce weight gain, and this lack of increased weight gain may track better with what's observed in the MAGEL2 null mice, which again don't gain more weight than wild-type counterparts, versus humans with PWS. Next, we turned our attention to evaluating the impact of MAGEL2 deficiency on food intake in rats. Overeating is a prominent feature of PWS, and it's thought that impairments in satiation contribute to that behavior. To test if MAGEL2 deficiency in rats produced hyperphagia and impaired satiation, we housed rats in a commercially available feedometer system similar to this one shown at the right. So in this caging system, the rats' food, which in this case is a standard chow diet, is located in a hopper that they have to kind of reach through a hole in the cage to get. And these hoppers sit on a small scale, this sort of blue box down here, that tracks feeding from the hopper on a moment-to-moment basis. So this gives us information in real time on food intake, but also meal patterns. And this is important because this provides information on behavioral mechanisms that might underlie changes in intake. So we examined meal size, which is a measure thought to reflect satiation processes, as well as the number of meals the animals took over the course of the day, which reflects satiety. And we hypothesized here that MAGEL2 deficient rats would have increased food intake, and that this would be due to an increase in meal size, reflecting impaired satiation in these animals. For these studies, we examined intake and meal patterns first at baseline, so sort of their day-to-day feeding. So this is baseline data from a particular day of their baseline week, day 6, just for sake of time, I picked a day. And this is data from our male rats. So the darker blue is the wild type, and the lighter blue, again, is the MAGEL2 deficient rats. So this is depicting cumulative meal-related chow intake over the course of a day. And we found no significant differences between the wild type and knockdown males for feeding. When we looked at meal pattern data, meal size, shown at the top right, again, thought to be a measure reflecting satiation, also did not differ between the two groups, nor did meal number at the bottom right, which again reflects satiety processes. So overall, these data did not support our hypothesis, and we're seeing that MAGEL2 deficiency did not impact this baseline feeding or meal patterns in male rats. Now, I mentioned we also did run female animals in these studies, and these are the day 6 baseline data from all the rats, both male and female. So the male data you're seeing here are the same as what you saw on the previous slide. However, the reason I'm showing these separately is with the data we've collected so far, we aren't able to run stats on the data from the female rats yet, because we only have data from one rat in the knockdown female group. And I'll explain why shortly. I just wanted to show you these data to give you a sense of kind of numerically what we're seeing. And we're also examining whether changes might be observed when these rats are presented with a challenge to energy balance. So not just their baseline intake, but when we give them a challenge. And for instance, we've looked at what happens if you deprive rats of food for 24 hours and then give them food back. So we've examined their intake and meal patterns after food is reintroduced after the deprivation period. I'm not showing you those data, again, just for sake of time, but in males, again, we found no differences in meal-related food intake, meal size, or meal number between the wild type and MAGEL2 deficient animals, suggesting that at least in males, MAGEL2 deficiency also isn't really impairing or changing their response to this challenge to normal energy balance. So I mentioned that we only have data from one animal in the female knockdown group for these studies, and we do have more animals, including female knockdown animals, that ran through these feedometer studies, but we had to exclude their data from our analyses because they exhibited hoarding behavior. So in other words, they kind of grabbed food out of the hopper and stashed it in their cage. And the problem is this precludes us from accurately measuring their food intake or their meal patterns. So we had to exclude them from these analyses, although we're continuing to run them in our further behavioral studies in our sort of battery of tests. But anyway, this was somewhat unexpected. So we've run cohorts of animals in these feedometer cages before. It's not uncommon to maybe have a rat or two that hoard that we have to exclude their data. But it just seemed that it was a lot more prevalent or predominant of a problem than we've seen previously. So we examined the percentage of rats in each sex genotype combination that exhibited hoarding on any of our feedometer testing days. And here's what we found. So in our wild type males, 14.3% of animals exhibited hoarding. And this is pretty consistent with what we've seen previously, so just a couple rats. In the knockdown males, almost 43% exhibited hoarding. In the female wild-type animals, about a third of animals exhibited hoarding. And then in the female knockdown group, so the MAGEL2 deficient females, nearly all of them, almost 93% of the animals exhibited hoarding. And this was really interesting to us. We ran some stats on the raw numbers of animals, like how many exhibited hoarding per condition, and did find an increased prevalence of hoarding in MAGEL2 knockdown animals. And hoarding has been observed in individuals with PWS. So it's a characteristic of this animal model, this hoarding behavior, that we'll be continuing to monitor and are interested in investigating in more detail in the future. So for the rest of the talk, I want to turn your attention to our next experimental question, which was whether MAGEL2 deficiency alters preference or motivation for palatable food. It's established that individuals with PWS have altered activity in neural systems relevant for feeding and motivation. And evaluations of food preference in PWS suggest that individuals may prefer sweet carbohydrate-rich foods. So we were interested in understanding whether MAGEL2 plays a role in food preference and/or the motivation for palatable foods. To test whether motivation to obtain palatable foods is altered in the context of MAGEL2 deficiency, we conducted self-administration studies where rats have to perform an operant task, in this case lever pressing, to obtain a small volume of a palatable fluid. And the fluids we used were either a 25% sucrose solution or a 10% intralipid solution. So sucrose being a carbohydrate-rich solution, intralipid being a fat emulsion, both palatable to rats. We opted to use liquids here because this allowed us to ensure that the reinforcers were isocaloric, so eliminating caloric density as a possible explanation for any differences in responding. For these studies, rats were trained to lever press for a small volume of either sucrose or intralipid on a progressive ratio schedule of reinforcement. So under this schedule, rats have to emit an increasing number of behavioral responses to obtain each successive reinforcer, so each successive bit of fluid. And this is thought to measure the animal's motivation to obtain the reinforcer. We measured 3 dependent variables: the active lever presses, the number of reinforcers earned, and the break point, or the largest ratio completed. And you can think of this kind of, this is the point at which the rats sort of give up. They throw in the towel, the response requirement has just become too high, and they're not willing to work for the fluid anymore. We hypothesized that MAGEL2 deficient rats would work harder to obtain palatable foods, reflecting an increased motivation, and that this difference would be especially pronounced for sucrose self-administration. So these are the data for sucrose self-administration in these animals, and the color scheme here for the males and females, knockdown wild type, is the same as you've seen before. Surprisingly, we didn't observe any significant differences in the outcomes evaluated. So active lever presses on the left, reinforcers earned, and then break point. This was contrary to our original hypothesis. Looking at these same outcome measures when animals were instead responding for fat solution, we did see some significant differences among groups. And specifically, we saw significant main effects of sex for active lever presses and for break point. So females were responding more for fat than were males. But there were no significant effects of genotype or sex-genotype interaction, suggesting that MAGEL2 deficiency isn't impacting motivation to obtain palatable foods in this test. So again, this is a work in progress. We're continuing to add subjects to these studies you've seen today, and we also have other experiments going on for these animals that we're working on. But to summarize what I've shown you today, MAGEL2 deficient rats did not exhibit increased weight gain compared to wild-type controls. We saw no major changes resulting from MAGEL2 deficiency for baseline food intake or, as I mentioned, refeeding in response to food deprivation in males. And the females is sort of an ongoing addition of subjects. There were sex differences, but no MAGEL2 genotype differences in our operant studies for fat solution, and no differences at all observed for responding for carbohydrate. We did observe hoarding quite frequently in our pedometer studies, and so it's possible that this may be useful to use MAGEL2 deficient rats to study this particular aspect of PWS. So as I mentioned, we're continuing to run more animals. We're running other studies to evaluate fluid preferences, as well as the neuronal responses to satiation signals in the hindbrain and hypothalamus of these animals. So please stay tuned for updates. I want to conclude by thanking those that helped with this work, so the members of my lab listed at the left. I'd also like to thank Dr. Rodney Samalko and his lab at Baylor, who provided us with the MAGEL2 deficient rats for these studies to start our breeding colony, and Dr. Derek Daniels and his lab at UB, the University of Buffalo, who are helping us with some of the fluid preference studies. These studies were supported by a grant from the Foundation for Prader-Willi Research. We're very grateful for their support. And unfortunately, I'm not able to attend the live question and answer session for the conference today, but I'm more than happy to answer questions. So please, if you want to discuss anything further or have questions about what you've seen today, my email address is on the screen. It's em1@buffalo.edu. So please feel free to reach out. And thank you for your time and attention.
Great. As you may have seen in the chat and in the video itself, unfortunately, we are not able to have a Q&A session for that presentation. You may also have noticed that that was not the presentation that I introduced, but we had a slight change, and we are now going to hear from Dr. Boscartiz. In addition, Dr. Yamada has been able to join us, and so following this last presentation, we will do back-to-back Q&A for Gabriela Bosco-Ortiz as well as for Dr. Yamada. So if you have questions for either of those presentations, please put them in the chat and we will do both of those following this next presentation. Thank you.
Hi, my name is Gabriela Bosco-Ortiz. I'm part of the Dietrich Lab at Yale University, and I'm happy to be here to share with you guys our findings looking at early behavior in MAGEL2 knockout mice. So taking a step back, MAGEL2 knockout mice are one of the many genetic models made for genes found in the chromosome 15 that contribute to the development of Prader-Willi syndrome. So these mouse models are useful to study the behavior in part because they have a homologous genetic region to chromosome 15 found in 7C. So out of these genes, we're looking at the MAGEL2 knockouts, which have been studied in the past and have shown to have impaired social behavior during adulthood. However, we know little about significant phenotypes in social behavior of this model during early ages. Part of the difficulty with studying behavior during earlier ages is that altricial mammals are born underdeveloped and they depend on their mothers or caregivers for all type of survival and nurturing needs. However, there is one behavior that is shared across mammalian species, and that is vocalization or crying. So here, this is the model, the behavior that we're going to be studying. This behavior is, as I said before, it's conserved between mammals. So even human babies are able to cry. And the cries are kind of used in the same way across species. They're meant to indicate a negative state, and they're in a very effective way to attract caretakers for nurture in infant pups. So the mice are particularly good at this behavior because it's very easy to induce and to manipulate in the laboratory setting. So infant vocalization in mice occurs whenever they're found in isolation. Here's an example of recordings that we did of their vocalizations while they were isolated. And we can see that whenever they're in reunion or in some kind of social context, vocalization rate always drops and reliably goes back up if the pups are isolated once again. So this behavior, in other words, is called an isolation-induced vocalization because it occurs during isolation. And because this is an infant behavior, it's most relevant during the first 2 weeks of life in the pups. And as the pups get older and more independent, they use the behavior less. So infant vocalizations, although they're widespread for mammals, in the case of mice, they're produced at the ultrasonic wavelength. So they're usually called ultrasonic vocalizations. And given that they're ultrasonic, we are not able to hear them, meaning that we have special tools to record these audios. And we also have a special in-house software called VocalMat to analyze not only how many vocalizations were made at the time, but also what the spectral features of these vocals looked like. And for the protocol in our tests, we decided to isolate pups from litters that contain both wild-type and MAGEL2 mutants. We would isolate them in an open field for 20 minutes while recording their vocalizations. As I said before, the vocalizations happen during the first 2 weeks of life. So we selected several ages during those first 2 weeks to look at the dynamics of the calls across ages. So now I'm going to get into our first set of results, looking at how many vocals were emitted during 20-minute isolation periods at different ages. And I'm going to go slowly through this because it will help understand some of the analysis done afterwards. So as you can see, at postnatal day 6, as you can see, the days that we tested were postnatal day 6, postnatal day 8, 10, and 12. If you first focus on the control mice, so the blue dots, you can see that the vocal behavior or vocal rate can be divided into 2 groups. There's the ages where there's a higher vocal rate, so which are the earlier ages, day 6 and day 8. And then there's the vocalization rate drops for days 10 and day 12. This pattern is not seen in the MAGEL2 mutants. They have high vocalization at P6, but vocalization drops at P8 and remain low for the subsequent ages tested. So the significant difference found in this case was at postnatal day 8. But we stay with the question about whether this is something that's peculiar to this age, or maybe it's because we see this drop on the behavior expression from earlier on in mutants versus controls. Besides looking at vocal rate, we looked at the spectral features, as I said before, and this is a diagram showing you how we measured different aspects of the vocalizations emitted by the pups. I'm just going to highlight where we found differences, which was in the maximum frequency or pitch made in the vocals, the bandwidth or the amplitude of the calls, and finally the duration, how long each of these calls lasted on average. So interestingly, we find that the differences only at postnatal day 8 once again, with the average vocalization being of lower frequency, maximum frequency, of a lower bandwidth, and of short duration. So given that the differences that we've outlined so far are very age specific, we wanted to find a way to answer the question as to whether this is just peculiar and unique to the age, or does it have to do with the shortening of the window for vocal behavior expression in the pups? And in order to do that, we used what's called a dimensionality reduction algorithm, which allows us to look at a lot of variables in a large data set and look for patterns or subsets of groups that would have been difficult to identify otherwise. So basically using this algorithm, specifically we used diffusion maps. We compared the spectral features of the vocalization of all the vocalizations emitted by the pups tested, and then we compared them between controls, so looking at the different ages, between the controls and between MAGEL2 mutant pups. So in the controls, we find again these 2 distinct behavior groups. So what we see specifically is that the spectral feature vocalizations of postnatal day 8 are very similar to those of postnatal day 6, whereas the features expressed at postnatal day 10 are very similar to those of day 12. So it seems like vocalizations, there's an expression seen at slightly younger ages versus the slightly older ages. But we don't see this like same clean break of two groups in the MAGEL2 mutant profile, where the postnatal day 8 actually doesn't vocalize very similarly to postnatal day 6 pups, and instead vocalize more similarly to postnatal day 10 and 12, so to the older pups. Finally, when we compare all of the ages and genotypes together, we further find results that suggest the same idea. So the MAGEL2 postnatal day 8 pups vocalize most similarly to control day 10 and day 12 pups. So it's not so much that the features highlighted during postnatal day 8 are just unique of that age, but rather that the expression of the behavior is somehow shortened in these pups in comparison to control. So they seem to not care for vocalizing from earlier on than the controls do, and which is an interesting feature that speaks to the defects in social behavior and bonding of these pups that might be manifested later on as autistic behavior phenotypes described in work from other works in the field. Finally, once we answered the question about what this means for the vocal behavior, so the idea that perhaps the behavior for this, the window for this behavior is shorter at this age, like we want to know whether that is meaningful in the pup's development at all. And in order to approach that question, we decided to execute a maternal retrieval test. So we have a mom in an open field with her nest in the middle of the open field, and we would put either a mutant pup or a control pup on each end of the open field and see which pups she would pick up first and how long she would take to pick up the pups and put them back in the nest. So this would help us understand if the mothers can somehow maternal behavior towards the pups can be influenced by this phenotypic difference that we found. This is an example of what the behavior, the maternal retrieval test looks like, where the mom is looking through the open field, and once she finds a pup, she will retrieve it back to her nest. So the results from our tests show that on average, that for the most part in most trials, moms would first pick up control pups and then pick up the MAGEL2 mutant pups. And then when we looked at the latency for retrieval, on average, it was longer for the MAGEL2 in comparison to control pups. And this is all at postnatal day 8. So we find that specifically at postnatal day 8, the moms prefer or prioritize the retrieval of control versus mutant pups. Interestingly, when we apply the same behavior, the same test at an earlier age, so maternal retrieval test with postnatal day 6 pups, we actually see that the retrieval latency between both groups is similar. There's not a significant difference in the time, and that kind of correlates to a time point where the vocalization profiles of both genotypes are similar as well. So this further suggests to us that that perhaps the postnatal day 8 pups are vocalizing more similarly to older pups that have less use for this behavior. And not only that, but it seems that the emission of this behavior affects the maternal care the pups receive in return. So to summarize the findings, the MAGL, we did see a robust differences in vocal behavior between, for the MAGL2 knockout pups, but we see it specifically at day 8. However, given the analysis that we did, we believe that it's not because of anything specific at postnatal day 8, and it's rather that the expression of this behavior is shorter and just overall less used during the development of the MAGL2 knockout pups in comparison to control. And we know that these features are relevant to the maternal care that they receive in return to the calls. So now we can say that the MAGL2 model has impaired behavior, social behavior in adulthood, but it also shows impaired social behavior and bonding at earlier ages in the neonatal stage of life. So with that, I would like to thank you for your time, and I'm happy to take any questions on this.
Thank you so much for that very interesting talk. We do have a question from Clementina, who is wondering if you have any insight as to what is happening during the brain development in that P6 to P8 range that could be impacting the vocalization difference that you're seeing.
I'm sorry, can you repeat what's happening at day 6, you said?
What's happening during brain development during P6 to P8? What do we know about what's occurring in brain development during that stage that would relate to vocalization?
Well, at these first few ages, the brain is like significantly growing by day. Like our coordinates to inject things in these pups, it differs basically by day. There's nothing particularly happening. Like at day 9 is when their ears fully open, for example. But there's not, besides the weight gain, like they're slightly bigger, there's not anything that could be changing. And the weights are similar, so yeah.
So where do you see this project then moving forward? Next steps.
Yeah, so that's a good question. Now that we know that there's this like behavior output, there's a behavior phenotype that we can measure at early ages, we can now start looking at circuits at these early ages. For example, in our lab, a project that I'm also involved in, I'm manipulating POMC neurons in the hypothalamus chemogenetically and hopefully also using optogenetics in the future to see how these neurons operate, for example, in social behavior for the pups. So knowing that we can study this phenotype, we can now use these same applications for MAGL2 mutants and start, for example, like doing calcium imaging in the brain if in different neural populations while they're in social reunion or in isolation, and try to get at like how these, especially like hypothalamic circuits of interest already in PWS, how are they performing in mouse models at younger ages? Does that make sense?
Mm-hmm. So Dr. Anthony Isles was commenting that one of the key changes that he's aware of that's occurring in that time period is maturation of the GABA inhibitory neurons. I
mean, maybe it's different in rats. I would just say that, like, by reading the literature, the development of the GABA receptors, like, varies across brain regions when it starts, but I had understood that that they were kind of developed as early as postnatal day 5.
Okay.
Like,
and then, oh, sorry, go ahead.
Yeah.
Rachel Wevrick was asking if you've considered trying oxytocin treatment, along the lines of some of Francoise Muscatelli's work, whether or not early oxytocin treatment might rescue some of this phenotype.
I'm very sure. I'd be pretty sure that it would, right? Based on like previous studies. There's a study now that's in preprint looking at latency for vocalization in MAGEL2 pups when they're in these like cold test protocols. And they do oxytocin and they see a rescue. So I think it would rescue. It's just that, like we're trying to think of like what more can we learn, right? Like, if I said it rescues, like what's happening in the mechanism. So we're really looking forward to better understanding like the circuit, like all the neurons involved in this behavior.
Then we have one more question, again from Anthony Isles. Are the maternal behaviors along the lines of the retrieval time, are they equivalent to those of 100% wild type litter mates, mums of 100% wild litter mates? And does the presence of the mutant pups affect the overall behavior level?
Well, I haven't done this retrieval test and just wild type mice, so I guess I couldn't compare. But that is an interesting question, because I think that the mother behavior could be affected by having the presence of MAGEL2 mutant and wild types in a same litter. And I've done studies with other strains where we do some cross-fostering data, and you can see that mothers are usually a bit more anxious. Yeah. It even affects the wild-type littermates' behaviors as well.
Excellent. Thank you so much. As I mentioned, we do have our first speaker here available now for Q&A. So I think we're going to bring him up onto the screen. And we did have a couple of questions that came in during your presentation. One was, did the ASD feature and AQ show up because most of your subjects were UPD? He wasn't sure if he missed what the breakdown was by genotype in your presentation. And this is from Dr. Ryder. Can you hear me, Doctor?
Hello?
Yeah. I'm sorry, were you able to hear my question?
Yeah, I'm sorry for your inconvenience.
Okay.
So could you please ask
Sure.
again?
Dr. Ryder was asking if the features you described, the ASD and the AQ, showed up because most of your subjects were UPD. He wasn't sure what the breakdown of genotype was. He wasn't sure if he missed it in your presentation.
So thanks so much for your point out the very important issue. So as you point out now, very important limitations of this study is that the control participants consisted of typically developing individuals and not those having the same level of intelligence. Moreover, so the participants consisted of only deletion group of genetic types. So I believe that we should be cautious about generalizing the results of our studies. So future comparative studies consisted of deletion group and UPD group are highly warranted. So yeah.
Okay. And then we have a question from Davis Ryman. He was wondering what difficulties you encountered trying to have the individuals with PWS complete fMRI, and how you were able to overcome some of those challenges. I think that any insight you can provide would be helpful to others who are trying to complete similar studies.
Mm-hmm. So most surprisingly, the individuals with PWS underwent the studies very successfully compared to those with Down syndrome or any other syndromes. So it may be beneficial for us to undergo these fMRI studies compared to other syndrome groups. So it was a very important and surprising aspect for us to better understand the characteristics of individuals with PWS and their behaviors.
Did you have anyone fall asleep in the machine or problems staying still?
I'm not sure exact reason for that. So I'd like to learn such aspects about and association with behavioral characteristics with PWS in the future.
Okay. And then the last question was whether or not you had any thoughts on what's happening at the molecular level.
So it is most important for us to understand more information between behavioral characteristics and genetic background. So I'd like to hear from any other researchers on this topic in the future.
Okay. Thank you so much for being able to join us for your Q&A, and thank you to all of our speakers in this session. I think there was a lot of fantastic information that was shared. Clearly, there's a lot of interest in all of these presentations based on the questions and comments that are coming in, and it seems like we may even have some opportunities for some new collaborations starting up, which is always very exciting. From our end at FPWR, we love it when we're able to bring researchers together to increase ideas and further research. We are going to have a short break now, so go grab a snack. I believe we have until where are we here? I believe we have until 1:40, so we have just shy of half an hour. I hope that you will take some of this time to go visit the poster session. I believe Caroline is going to include a link to the posters as well as to the YouTube channel that has the recorded presentation, the walkthrough presentations for the poster sessions. So I hope that you'll take some of this time to view those, and then we will see everybody back here in just under half an hour. Thank you again.
Well, welcome back, everybody, to the final session of this year's research symposium. Hopefully everybody got a nice snack and we're all refreshed for this last group of speakers. So we'll start off by looking at some work from Françoise Muscatelli and Sebastian Boret. And these investigators work closely together. And so I think what we're going to do is have Françoise present her talk, and then Sebastian present his talk. And then at the end, we'll have both of them join us for some questions. So Françoise's talk is silencing oxytocin neurons during critical developmental timings induces long-term behavioral alterations similar to alterations of MAGEL2 deficient mice. And Sebastian Boret will be talking about how oxytocin influences the development of the melanocortin system and adiposity during distinct periods of postnatal life.
I'm Françoise Muscatelli, and I work in France at Marseille at the Institute of Neurobiology of Mediterranean, that you can see here. And I work for many years now on Prader-Willi syndrome, and specifically on genes involved in Prader-Willi syndrome, like in mouse models, like MAGEL2 knockout mouse model and the Negin knockout mouse model. So today I'm going to present. First, I would like to thank the organizers for giving me the opportunity to present our work today. And I'm going to present the study that has been granted by FPWR. And it's a large study involving Sebastian Boret and myself. And in my team, we have created the mouse models to silence OT neurons during critical developmental timings to study the effect of this transient silencing on adult behavior. So MAGEL2, as you know, is a gene involved in Prader-Willi syndrome and Scharf-Yang syndrome. And previously, we have shown that the inactivation of MAGEL2 gene in mouse is responsible for the deficit of sucking in pups. And it's why they die after birth, at least part of them. And in adolescence and adulthood, the knockout mouse model present an altered behavior, social behavior, and an altered cognition. And we have shown that the administration of oxytocin during the first week of life save the pups. They do suckle properly, and at the adult age, they do present a normal behavior. So, why this project? It's to prove that oxytocin can shape the structure of the nervous system in a critical window of time and as a result impacts the adult animal behavior. So what's a critical period? It's something that it must happen, or it either won't happen, or it is problematic if it doesn't happen. And a critical time window can be easily understood by looking at the cycle of a plant. If one of these developmental stage at one specific time during spring or summer or autumn, if something is wrong, the plant is going to die or is not going to develop properly. So the consequences of this project is to try to determine the most appropriate window of time to administrate oxytocin in patients. So oxytocin neurons have a dynamic process from after birth during development, from P0, P means the day at birth, until the adulthood. The expression of oxytocin receptors is also a dynamic process during development. So to inactivate oxytocin neurons transiently, we have used the DREAD for designer receptors exclusively activated by designer drugs technique. And briefly, what we have done is just we used an oxytocin cream mice. It means that an oxytocin, a transgenic mouse expressing the queer recombinase protein under the oxytocin promoter with a transgenic mouse carrying a DREAD, and this DREAD is able to inactivate neurons. By this crossing, we have the expression of oxytocin Cre and the DREAD, which is not inactivated activated yet, in the oxytocin neurons. And the DREAD sequence is flanked by LOXP site, and with the query recombinase, we are going to have the DWED expression. So this expression of this receptor at the surface of oxytocin neurons is going to be activated by a specific inert ligand, CNO or compound 21. And like that, we can silence the oxytocin neuron specifically during the time where compound 21 is present. So compound 21 is able just to inactivate for 6 hours. So we do different administration, just like subcutaneous injection in the animals to inactivate for several hours or several days. So just Cre plus mice are the mice in which oxytocin neurons are inactivated, and Cre minus mice are the control mice, and they are the best control because we can administrate compound 21 to see the adverse effect, the possible adverse effect, and in the control mice. So we choose to inactivate oxytocin neurons at 4 critical stages, at the day of birth only, or during the first week of life from P0 E7, or during 1 week in early adolescence, or during 1 week in young adult. And we study here the behavior. Sébastien is going to present the work on metabolism and hypothalamic neurodevelopment. So we have performed different behavioral experiments. First, ultrasonic vocalization in pups, 8 days after birth. And why? Because ultrasonic vocalization in pups is the first social communication. The USV are produced by pups when they are outside the nest and far from the mother. And these calls are defined as isolation and USV. We performed also the Rotarod test. This test is done just to test motor coordination, balance, muscle strength, and fitness. And mice are placed on a rotating rod, and latency until the mice fall from the rod are either constant or accelerating speed can be detected. The elevated plus mice is an anxiety test and is based on the response of mice to a potential aversive environment. The mouse is placed in the center zone where the forearms meet and allowed to explore freely the open arms or the closed arms. The open arms are considered just like an aversive environment, and the closed arms are considered as a safe environment. So increased anxiety leads to a shorter duration of time spent in the open arms. We also take the spontaneous activity, so we look at the movement, the space occupied, the wearings of the grooming behavior, and this image, for instance, is an example of movement for a normal mouse, and this is the image represents the movement when you have an autistic mouse. The novel object recognition test allows to test the interest of a mouse for an object and for a novel object, revealing the curiosity and interest for the environment. An autistic mouse will not spend more time with a novel object compared with the old object. Finally, we test using an open field the spontaneous interaction, and this is a test for sociability. We use an open field equipment and place the test mouse together with an unfamiliar mouse into the same arena and evaluate various parameters of social interaction, such as the number of sniffing between mice. The three-chamber test is a more complex test. It's done at 4 months old. And what we do is we place a mouse in the center. We have 3 chambers, just like represented here. And first, the habituation trial, the test mouse is placed in the central part of the 3-chamber unit with 2 empty wire cages located in the left and right chambers. The mouse is allowed to fully explore each chamber here. And 5 minutes later, we put a novel mouse in one of the chambers, and the normal mouse is going to spend time just to explore this novel mouse, just like air. And 5 minutes later, we add a second mouse in the other chamber. And again, a control mouse is going to spend more time to explore the novel mouse. And 30 minutes later, we test the short-term social memory, adding a third novel mouse in the chamber in another place. And we should observe in a control mouse much more time to spend to explore the new novel mouse compared with the familiar mouse. Having said that, we have used all those experiments to assess the behavior in different mice. First, the first cohort of mice in which we have just inactivated oxytocin neurons just 1 day after birth, but we didn't observe any significant effect. And the second cohort of mice, we have administered compound 21 to inactivate oxytocin neurons during the first week of life. And what we observed is represented in the different slides. So concerning the ultrasonic vocalizations, we observed a reduction in the number of ultrasonic vocalization in the Crepus, sorry, this is not in the good order, in the Crepus mice. So there is a decrease of USV number at P10 following a silencing of oxytocin neurons during the first week of life, just like we observed in MAGEL2 knockout phenotype. Concerning the motor activity, we observed a tendency to have less activity in females, but this is not really significant. Concerning the global activity evaluated in an open field, we observed that in the mutant mice, there is an alteration in the trajectory and occupied space, with, in fact, similar time spent in the border or in the center. We also observed in male an alteration of the grooming activity with an increase of grooming activity in males and a decrease in females following the inactivation during this first week of life. Concerning the novel object recognition test, we observed no discrimination between the old and novel object in male and female CRE-positive mice. And we also observe total time interacting with the novel or even the same object, which is reduced in the mice in which the neurons have been, the oxytocin neurons have been activated during the first week of life. Looking at the spontaneous social interaction and specifically at the specific time between both mice, we observe that there is less spontaneous interaction in CRE plus male mice. This is a tendency, not significant. Finally, looking at the social interaction with the three-chamber test, we observed in the CRE plus mice loss of short-term social memory. So the first conclusion is that we observed after the inhibition of oxytocin neurons during the first week of life, long-term effects on social behavior with a normal motor activity, but less USV when pups are separated from the mother, an impairment of occupied space and grooming, an impairment of no, less time to interact with the object, less spontaneous social interaction, and loss of social memory. So the effects we have shown, I didn't present that very precisely, are different in males and females, and this is expected. And all those alterations are also present in the MAGEL2 knockout mouse model, as we have shown previously. So then in the third cohort, we have inactivated the oxytocin neurons during 1 week in adolescence. And I'm just going to summarize the results. We did not observe any effect on USV because we have not tested USV. It should be tested at P8, 8 days after birth or 10 days after birth. The motor activity is normal, but we have observed definitely an impairment in the novel object recognition test with male CRE plus animals interact significantly less with the object, familiar or new object. we have observed less spontaneous interaction in mates and also an impairment of social memory. The elevated plus mice results are still variable, just like before, and difficult to interpret. This is just to show the decrease of time to interact between two mice, Equiplus and the novel stranger mouse, compared with the Cre minus males. Finally, we have inactivated the oxytocin neurons in young adult mice, so between P56 and P62, and we are still producing some results, but now we have shown that, okay, USV are not tested because we do not have PEPs. Their motor activity is normal, but we have observed an impairment in the novel object recognition test, and this was quite not unexpected, and the malcripus animals interact significantly less with the object, familiar or just like we observe at the adolescent stage. Other experiments are now ongoing. So in conclusion, I would say that definitely transient inactivation of oxytocin neurons in the first week of life or during adolescence and putatively in adulthood has long-term effects, altering the social behavior. The behavioral alterations we observed are similar to those found in autistic-like mice, already described in several articles. Those results suggest that administration of oxytocin during those periods can rescue the phenotype with long-term effects. And definitely we observe that alterations found in MAGEL2 knockout mice can be rescued, except USV, by an administration of oxytocin in the first week of life. So we think that the administration of oxytocin in MAGEL2 knockout during adolescence should have similar effects. So now we would like to acknowledge people in my team, Fabienne Schaller and Stéphane Gaillard, and our collaborators, Sébastien Bourret, Pierre-Yves Barrel, and Sébastien is going to present the work on those mouse concerning the metabolism and the hypothalamic development. So maybe it's time for questions. So please go ahead with questions. Thank you very much.
Hello, everyone. I'm Sébastien Bourret, and I'm a research director at the Institute Neuroscience and Cognition in Lille, France. And today I'm going to present you the FPWR funded projects that we have with Françoise Muscatelli in Marseille. And this project aims at studying the importance of oxytocin during different periods of life on the development of the melanocortin system and adiposity. So as Françoise mentioned just before me, oxytocin neurons play an important role in social behavior and cognition, but they also play a critical role in metabolic regulation. And one of the reasons why oxytocin neurons play such an important role in metabolic regulation is because they can interact with the melanocortin system in the arcuate nucleus of the hypothalamus. And the melanocortin system is composed of neurons that produce pro-opiomelanocortin, or POMC, but also neurons that produce agouti-related peptide, or AGRP, and neuropeptidoid, or NPOI. And POMC neurons are anorexigenic, so they decrease food intake, whereas AGRP, NPOI neurons are orexigenic, so they increase food intake when they are stimulated. So my lab is interested in studying how hypothalamic circuits that regulate feeding and metabolism develop. And what we know is that the melanocortin system begins to develop during mid-gestation, around embryonic day 11 in mice, but the vast majority of the development of the melanocortin system occurs postnatally during the first 2 weeks of postnatal life in mice and rats, with the development of POMC and AGRP NPOI axonal projections. And what's very interesting to see is that just prior to the development of the melanocortin system, there is a postnatal maturation of oxytocin neurons, suggesting that oxytocin might actually play a developmental role on the melanocortin system. So therefore, the two research questions that we had were to explore whether oxytocin exerts neurodevelopmental effects on hypothalamic melanocortin circuits, but also to examine whether there is a critical period during which oxytocin exerts maximal effects on the development of melanocortin circuits. So if oxytocin is very important for the postnatal maturation of the hypothalamus, then the oxytocin receptor should be expressed in the hypothalamus during development. So for that reason, we first examine the mRNA expression of oxytocin receptors in various nuclei of the hypothalamus on postnatal day 10, which is a critical window of hypothalamic development. And we found that oxytocin receptor mRNA was expressed throughout the hypothalamus, and particularly in the arcuate nucleus of the hypothalamus, where POMC and AGRP and PY neurons are located. To further determine whether oxytocin might actually play a neurotrophic effect, we then performed ex vivo experiment where we took explants from the arcuate nucleus, incubated them with oxytocin, and looked at axon growth. And we found that oxytocin could indeed promote axon growth from the arcuate nucleus of the hypothalamus, supporting the idea that oxytocin does play a role in hypothalamic development. But obviously, these data were from in vitro experiments, and it was very important to confirm whether there was a neurotrophic effect of oxytocin in vivo and what was the physiological relevance of these findings. So therefore, we used the mouse model that Françoise developed and that Françoise just described to you. So basically, we used a mouse model in which oxytocin neurons express the GI DREAD HM4 DI receptors, so that when mice were injected, or when these mice were injected with the DREAD compound or the DREAD ligand compound 21, then oxytocin neurons were inhibited. And what Françoise and Fabienne Schaller did was to inject compound 21, so to inhibit chemogenetically oxytocin neurons either during the neonatal life, so between birth and postnatal day 7, or during the juvenile period, so between postnatal day 25 and postnatal day 32, or during the adult life, so between postnatal day 56 and 62. And then several weeks later, we took tissues from these mice to look at adult outcomes, which were the examination of hypothalamic development, and also we've looked at body weight and adiposity. So today I'm just going to present you what happens when you inhibit oxytocin neurons either during the neonatal life or juvenile life, because we're still collecting data from the adult experiments. So what happens when we look at the brain of these mice? So these are axonal projections from the arcuate nucleus to the paraventricular nucleus. And these are axonal projections that contain pro-opiomelanocortin. And what we found is that when we inhibit oxytocin neurons neonatally, then we disrupt the development of axonal projections, POMC axonal projections from the arcuate nucleus to the paraventricular nucleus. But when oxytocin neurons were inhibited during the juvenile period, we didn't have any impact on POMC projections. Similarly, if we inhibit oxytocin neurons during the neonatal period, we reduced the density of AGRP projections to the paraventricular nucleus of the hypothalamus. But if we inhibit oxytocin neurons during the juvenile period, we didn't have any impact on AGRP projections to the PVH. Very interestingly, we found that this inhibitory or this effect of oxytocin inhibition on POMC and AGRP projection was only seen in male mice, because POMC and AGRP projections to the PVH were completely normal in female mice. We've also looked at other projection pathways of POMC and AGRP projections, and particularly to projections to the dorsomedial nucleus of the hypothalamus. But in contrast to POMC and AGRP projections to the paraventricular nucleus, projections to the DMH were completely normal, whether oxytocin neurons were inhibited during the neonatal life or during the juvenile life, and that both in males and female mice. We then wanted to know whether the inhibition of oxytocin neurons during distinct critical developmental periods could have an impact on the physiology of the animal. So we've measured the body weight of the mice. And what we found is that the neonatal inhibition of oxytocin neurons did not have any impact on body weight, whether it's in males or in females. However, if we inhibited oxytocin neurons during the juvenile period, then the animals were heavier, but only the male mice. The female mice were completely normal as well. So one of the reasons why the animals were heavier is because they were fatter. So we measured the fat mass of mice that had oxytocin neurons inhibited. And what we found is that the juvenile inhibition of oxytocin neurons resulted in an increased fat mass. And this increased fat mass was associated with bigger adipocytes. And actually, if we look carefully at the adipocyte distribution, what we found is that mice that had oxytocin neurons inhibited during the juvenile life had less adipocytes of small size, but more adipocytes of bigger size. So clearly, we had a very strong impact on adiposity when we inhibited oxytocin neurons during the juvenile life. And that's very interesting because there was a paper published a couple of years ago that suggested that oxytocin actually might play a direct role in adipocyte development and maturation. So what this paper found, and was published in PNAS in 2019, was that oxytocin receptor was expressed in the adipose tissue, and that actually, in fact, the adipose tissue was the organ that expressed the highest level of oxytocin receptor mRNA. And then what they found is that in mice that do not express oxytocin receptor in the entire body, these mice were fatter, so with an increased fat mass, particularly in male mice. So that suggests that oxytocin might actually play a direct role in in the adipose tissue development and maturation. And what our results indicates is that this developmental action of oxytocin on the adipose tissue might specifically occur during the juvenile life. So of course, we are here at the FPWR meetings. So the question is, what is the relevance of our findings for PWS? And what we do know is that patients and actually mouse model of PWS have a loss of oxytocin neurons. So there is a deficiency in oxytocin, particularly during early life, either in patients with PWS or also in a mouse model of PWS. And what we found a few years ago was that the development of POMC projection was disrupted in a mouse model of PWS, which is a MAGEL2 null mice, but that AGRP projections were completely normal in MAGEL2 null mice. So suggesting that there might be a link between the loss of oxytocin neurons with the disrupted development of POMC projections that we see in MAGEL2 null mice. So to further address this question, we used another mouse model that Françoise developed, but this time we used a MAGEL2 null mice in which oxytocin neurons expressed the HM3DQ DREAD receptor, so that when we injected the compound 21, which is a ligand of the DREAD, then oxytocin neurons were stimulated. So they were not inhibited, but they were stimulated. And then what Françoise did was to inject the compound 21 between postnatal day 0 and postnatal day 7. And then a few weeks later, we've looked at the hypothalamic development and body weight and adiposity. And what we found is that in MAGEL2 null mice that had oxytocin neurons stimulated during early life, then we had actually an increased density of POMC projection innervating the paraventricular nucleus of the hypothalamus, only in male mice, but not in female mice. And that AGRP projections were completely normal in both control and knockout or mutant mice. Then we've also looked at the body weight and the adiposity of control mice, and in mice in which oxytocin neurons were stimulated during early life, and these again have a MAGEL2 null background, and we didn't see any impact on body weight or adiposity, whether it's male or female MAGEL2 null mice that had oxytocin neurons stimulated during early life. So to summarize, what we found was that oxytocin receptor mRNA is expressed during critical periods of hypothalamic development, that oxytocin can at least act directly at the level of the hypothalamus to promote axon growth. And then when we inhibit oxytocin neurons during early life, so between birth and postnatal age 7, then we can impact hypothalamic development, and particularly the development of projections that contain POMC and HRP. And this was very specific to the projections to the PVH, because projections to the PVH were completely normal. And then we didn't have any effect on body weight and adiposity. But in contrast, if we inhibit oxytocin neurons during the juvenile period, then we did not impact hypothalamic development, but we did have an effect on body weight and adiposity. And that might be actually the result of a direct effect of oxytocin on adipose tissue development. So that's very interesting, because it tells you that depending on when you are acting, whether it's during early life or during the juvenile life, you might actually modulate a very different developmental program and either affect hypothalamic development or adipose tissue development. And then what we also found is that in a mouse model of PWS, which is a MAGEL2 null mice, when you stimulate oxytocin neurons during early life, then you can actually reverse the abnormal POMC axonal projection development that we found previously in this mouse model. Finally, I would like to thank people of my lab, and particularly my PhD student, Pierre-Yves Barrel, who did all the work, neuroanatomical work and metabolic work in both the DREAD inhibitory and the MAGEL2 DQ mice. And also my collaborator, Françoise Muscatelli, and our technician, Fabienne, who generated and provided us with the mice. And of course, I would like to thank very much the FPWR and the families who provided support for this very interesting work, which hopefully would have an impact for treatments. And I'd be very happy to answer questions if you have any. Thank you.
Great. Thank you so much, Françoise and Sebastian, for that great work and that great collaboration. We do have a few questions coming in. As a reminder, please add your questions to the chat box. The first one is from Clementina, asking about would child neglect in early life resemble the inhibition of the oxytocin neurons?
Sorry, I didn't hear the question. What was the question?
Sorry, the question was, do you think that early neglect, early in life in childhood would resemble inhibition of the oxytocin neurons?
I think that oxytocin neurons inhibition, at least yes, mainly inhibition, play a role in Prader-Willi syndrome and in childhood. What is important to know is that in MAGEL2 deficient mice, there is a deficit of oxytocin, mature oxytocin, and release of mature oxytocin in early life. But later on, at adulthood, at least in terms of quantity and amount of oxytocin, the amount appears quite normal, but possibly the oxytocin circuitry should be affected.
And I think the question was getting at in sort of the wild type or normal situation, if you were to reduce oxytocin, I mean, are there environmental factors that can be also impacting oxytocin and development? I think it's just a general question.
Well, generally, yes. I mean, environmental factors can impact oxytocin neurons, just like stress. And this has been described. If you stress the pups in wild-type mice, and you isolate pups from the mother for 1-2 hours every day, you're going to have a decrease of oxytocin release, and you're going to impact the social behavior as it is impacted when you have a deficit of oxytocin.
And actually, if I can add something also, maternal obesity has been associated with the loss of oxytocin neurons in the offspring. And very interestingly, if you stimulate oxytocin neurons in pups born to obese mothers with some microbiota, then you can correct the social behavior that is defective in pups born to obese mother. So there is another environmental influence of oxytocin production.
So in theory, could you... go ahead. I was going to say, in theory, if you had a prenatal diagnosis of PWS, could you be, giving mom something that would help the oxytocin neurons develop more normally?
No, because oxytocin in pups is released from birth. And oxytocin in the mother is something. Oxytocin in the pups is something else. So it's different things.
Great. All right, and we had a question from Marnie Blewett about the in some of the later timings of oxytocin neuron inactivation. You mentioned that the male mice have an effect, and the question is, were the females unaffected, or what was the sex difference?
The point is that we use the three-chamber test in those tests, and we have published recently that the three-chamber test is okay when you test the males, just to look at the social deficiency or the social interaction, and specifically for social memory that is known to be disturbed in oxytocin-knockout mice, for instance. But the three-chamber test is not appropriate to test females, and many tests have been performed and have been tested in males mainly. Females have been really, there are few studies on females, honestly, and I think that most of the tests are not appropriate for females. But definitely in the open field, looking at the spontaneous interaction or the spontaneous behaviors of females, we didn't observe the same things.
Okay, thank you. And I guess I had a question about the effects of the central versus the peripheral from your work, Sebastian. I mean, so do you think there is a potential therapeutically, separately to administer oxytocin peripherally versus centrally, or?
It actually would be very interesting, because when we chemogenetically inhibit oxytocin neurons, then we end up having also loss of oxytocin production in the portal blood as well. So we have both a central and a peripheral effect, but it would be interesting maybe to inject oxytocin peripherally or an antagonist peripherally to see if we can just target the adiposity. That could be interesting too.
Yes, but it's difficult to do because, when you do inject peripherally oxytocin, that's what we do anyway. We do perform, in our previous articles, peripheral injections. The idea is that you have a small quantity of oxytocin that is going in the brain. And this has been shown. So it's difficult to have a specific action in periphery and not in the brain.
Yes, yes. All right, I don't see any additional questions from our audience, so I will thank you both for your work and for the discussion. And we will move on to our next presentation, which is from Kritika Bala, looking at the functional characterization of PrEPL, a serine hydrolase deleted in a Prader-Willi-like syndrome.
So thank you very much. In 1966, two medics from Antwerp, Clara and Lowenthal, they published first 3 patients with PrEPL deficiency. At that time, no genetics was detected, but what they did notice was similarity to Prader-Willi-like syndrome, like generalized neonatal hypertonia, growth hormone deficiency, failure to thrive, and later during the adulthood, hyperphagia and rapid weight gain. At that stage, they did suspect some molecular links with Prader-Willi syndrome, but only recently we have been able to establish those links that I'm going to talk about in the next 15 minutes. So first I'll be giving introduction about CMS22 or PREPL deficiency, and then I'll be talking about the expression of PREPL in the tissues of mouse model for Prader-Willi syndrome. And finally, I'll be talking about how mitochondrial function is affected in our PREPL knockout mouse model. So inactivation of prylyl endopeptidase-like gene on 2p21 chromosome leads to novel recessive disorder called congenital myasthenic syndrome 22. Until now, no drug has been able to fully treat this disorder, and hence it is of prior importance to look deeper into the molecular mechanisms. Next, I would like to talk about another neurodevelopmental disorder called Prader-Willi-like syndrome. So CMS22 shares phenotypic similarities with this syndrome, starting from neonatal hypotonia, feeding problems in child, leading to failure to thrive, and also later during the adult stages, the hypophagic character and also obesity. Apart from that, cognitive and growth impairment is also observed, and infants also do have hypergonadotropic hypogonadism. Now, difference between these two disorders is that firstly, in Prader-Willi syndrome, the symptoms are pretty severe. For example, obsessive compulsive behavior is observed in these infants like skin picking, and also there is problem with the thermoregulation. Apart from that, eyelid ptosis is very frequently observed in CMS22, which is not the case in Prader-Willi-like syndrome. So these similarities are striking, and all initially the CMS22 patients were first diagnosed with PWS phenotype without the genotype, and hence we hypothesize that the overlapping pathways are affected. So PREPL, it is a serine hydrolase which belongs to oligopeptidase family. It has two isoforms, PREPL short form which encodes for 638 amino acids and PREPL long form which encodes for 727 amino acids. Recently, in collaboration, crystal structure of PREPL was solved, and it consists of two domains, the catalytic domain which is made up of alpha-beta hydrolase fold which is connected to a beta-propeller domain via hinge. The catalytic domain has catalytic triad residues which are responsible for the hydrolase activity of PREPL. Now, PREPL shares 26% of sequence similarity with its only human ortholog, that is PrEP, and PrEP itself is a serine protease. So initially, all the secondary structure elements were known to be conserved in PREPL and PrEP, and also these catalytic triad residues, they're present at topologically equivalent position in both of them. Hence, a peptidase activity was expected. But until now, we haven't been able to find any peptide substrates. But recently, we discovered that PREPL has a thioesterase activity, and we were able to confirm its esterase activity using p-nitrophenyl esters of variable length and thioesterase activity using p-nitrophenyl thioacetate. These are the only substrates found for PREPL so far. Next, PREPL is a protein which is ubiquitously expressed with its highest expression found in brain and intermediary levels in kidney, muscle, and neuroendocrine cells. So immunofluorescence, a MAPIT study was performed by a lab, and where we found out that 32% of PREPL interactome was found to be linked to mitochondria. So based on that, immunofluorescence study was performed, and as you can see here clearly that PREPL can be seen localized with mitotracker. But its localization was not only just limited to mitochondria. The mitochondrial fractions were separated from cytosolic fractions, and what we can see here is that PREPL can also be found in cytosol. Interestingly, long form of PREPL is residing in mitochondria while the short form resides in the cytoplasm. COX-4 is the nuclear encoded subunit of complex 4, and here is used as a mitochondrial marker. So until now, what do we know about PREPL is that it is a thioesterase and not peptidase, and somehow involved in mitochondrial functioning. Since it shares phenotypic similarities with Prader-Willi-like syndrome, we hypothesize that the PREPL deficiency is the underlying cause of the neonatal hypotonia and growth hormone deficiency that we see in Prader-Willi patients. So we wanted to check expression level of PREPL in tissues for mouse model for Prader-Willi syndrome. And on this project, we have been collaborating with Professor James Resnick, who was kind enough to provide tissues for the study. So firstly, we are using this PWS IC deleted mutant mice, and in these mice, there is a targeted deletion of 35 kb that includes this imprinting center region and exons 1 to 6 of SNRPN, which results in the abolishment of expression of all the paternally imprinted genes. Now, it is a very lethal mouse model with the mice maximum surviving up to 7 days. And also they are really small, they have lower body weight, they show neonatal hypotonia, they fail to thrive, and also the decreased movements are present. So basically all the phenotypes that we are interested in Prader-Willi syndrome can be studied well in this mouse model. So next mouse model we chose is a Prader knockout mouse model. This model has been generated and well characterized in our lab. Exon tail of Prader has been deleted in this mouse model, and because of that, it is a frameshift mutation which results in the loss of all the catalytic triad residues and hence the hydrolase activity of Prader. Apart from that, there is also no residual protein expression of Prader because of a premature stop codon. Also, these mice, they die perinatally within the first hour of their birth. They also have reduced size and body weight, as you can see here when you compare wild type versus knockout, and also they're hypotonic and they have weak muscle tone. So again, a very good mouse model for study. So, but why Prader? Prader is known to be 2.5 times downregulated in brain of Prader-Willi patients and also 4 to 16 times in the muscle biopsies. So that's why we just decided to check the expression of levels of Prader in this PWS IC-deleted mouse model. And clearly, a prominent 4-fold reduction can be seen in a Prader-Willi mice compared to the wild type. And now in these two graphs, in the first one, we actually use two different primer pair sets. In first, this primer pair specifically binds to the long, that is the mitochondrial form of Prader, while in second, it binds to both long and the short, that is the cytoplasmic form. So Prader is downregulated in Prader-Willi mouse model, and then we decided to check the expression level of PCSK1 and its upstream regulator, that is NHLH2. Now, PCSK1 encodes a protein or enzyme called PC1, which is known to convert the proteins into their mature hormones or neuropeptides, some of them which are also known to regulate food intake, body weight, and growth. And why are we interested in this enzyme? Because according to previous reports, the expression of PCSK1 and its upstream regulator, they have been found to be reduced in induced pluripotent stem cell-derived neuron of Prader-Willi patients and also some tissues of Prader-Willi mouse models. There was another one report that did not detect any differences in both of them, so we decided to look into the controversial role of this PCSK1, and what we found out is downregulation not only in Prader-Willi mouse model but also Prader knockout mouse model. Apart from that, we also found out that there is a 40% reduction of PC1 expression in 3 different Prader knockdown beta TC3 cells. Collectively, which suggests that PC1 function is impaired in both the mouse models and serve as a contributing factor, not only in PWS but CMS22 phenotype. Surprisingly, these neonatal Prader knockout and PWS mice, they showed a reverse trend of upregulation of this NHLH2 expressions in the brain. So our data of NHLH2 is highly controversial. Next, as I have mentioned before, that both the mouse models, they display feeding problems and also postnatal growth retardation. So we decided to check first the feeding problems, and for that we wanted to see the expression levels of orexogenic neuropeptide. Clearly, we can see that the expression levels of this primary appetite stimulating neuropeptide or NPY or neuropeptide Y is reduced in both the mouse models. So it seems like that these mice, they're hungry, but they're not getting the signal to eat, suggesting a dysfunctional or exogenic signaling, which leads to failure in thrive in both the mouse models. Next, for postnatal growth retardation, we specifically looked into this growth hormone secretor receptor or ghrelin receptor. And why is that? Firstly, this receptor is known to regulate hypothalamic pituitary signaling, which stimulates the growth hormone release from the pituitary. Secondly, this receptor is also known to regulate the food intake by forming heteromers with dopamine receptor in specific subset of hypothalamic neurons. Apart from that, in midbrain and hippocampus, this receptor plays an important role in learning and memory and also has neuroprotective effects. So since all of these features are altered in PWS and CMS22 patients, we decided to look at expression levels as well. And clearly we see a downregulation, strong downregulation in the brains of both Prader-Willi and triple knockout mice. We also check the expression levels of this POMC neuropeptide or pro-opiomelanocortin neuropeptide, which is a satiety hormone which remain unaltered in both the mouse models that suggest the unaffected and or exogenic signaling in both of them. Altogether, these data uncover very strong molecular link with Prader-Willi-like syndrome and also PREPL deficiency. Coming to the final mitochondrial function, firstly, I would like to talk about these two papers. In this first study, Yazdi mentions that the oxidative phosphorylation complex activities, they're impaired in both brain and muscle of Prader-Willi IC-deleted mice. Apart from that, this prelim study by Butler showed that the mitochondrial bioenergetics is also disturbed in Prader-Willi patients. And interestingly, isolated complex 4 as well as combined complex 4 deficiencies have also been observed in several PREPL deficient patients. And I also mentioned before that we found out that the 32% of PREPL interactome was linked to mitochondria, with significant number of mitochondrial proteins that are involved in oxidative phosphorylation and mitochondrial gene expression. So that's why we decided to check the effect of PREPL deficiency on mitochondrial respiratory complex activities. We checked this in cerebellum, and the activities were normalized to citrate synthase. And clearly you can see a downregulation in the activity of complex 1, 3, and 4, while the activity of complex 2 remain unaltered. So now complex 1, 3, and 4, they're encoded by mitochondrial DNA, and complex 2 is encoded by nuclear DNA. We also checked these in cortex and quadriceps of PREPL knockout mice, and we also saw the similar trend of downregulation in complexes. Next, the 15 PREPL interactors are the proteins which are also involved in mitoribosome function. So based on that, we also decided to check the effect of PREPL deficiency on protein synthesis or protein translation. And clearly we can see here a decreased protein levels of this cytochrome B, which is encoded by mitochondrial DNA, in the quadriceps of PREPL knockout mice, while the protein expression of nuclear-encoded COX-4 remained unaltered, suggesting only mitochondrial but not nuclear translation is disturbed in PREPL knockout mice. So all in all, this data surely reveals that PREPL is involved in mitochondrial dysfunction. Apart from that, we also studied the electron micrographs of these quadriceps of PREPL knockout mice, which revealed the abnormal morphology of mitochondria. We can see the mitochondria swollen also with the loss of cristae. These cristae are less organized and also frequently dilated. So again, suggesting PREPL has a very important role in mitochondrial function. So now coming to the conclusions, first is the PREPL is a thioesterase which is localized in mitochondria. It is downregulated in PWS mouse model, and it is also involved in mitochondrial translation and respiratory chain functions. Our future experiments include rescuing of this neonatal lethality in PWS IC-deleted mice and also the mitochondrial phenotype by expressing transgenic PREPL. Again, we will be collaborating with Professor James Resnick on this project, and this project really has the capability to unravel many therapeutic avenues, not only for the treatment of Prader-Willi patients, but also CMS22 patients. At the end, I would like to thank you, Professor John Krimmers and our whole team, also our collaborators, and the Lacroix Funds and FWO for supporting this study. Next, I'll be happy to answer the few questions now. Thank you.
Great, thank you for that talk. We have a couple of questions coming in. excuse me. The first is, why do you believe that the mitochondria are disturbed in the PrEPL knockout, but not the nuclear translation? I guess the different, the short and the long.
So the mitochondrial translation, of course, is affected, as we have seen, and the reason is because we think that PrEPL long form is involved, and PrEPL is not found in the nucleus or seems to have a role there, but it is present in cytoplasm where it does the short form. The short form is present in cytoplasm where it is known to be involved in regulatory secretion, and is basically known to disrupt the vesicle trafficking pathway. That's why we think that nuclear DNA is not, nuclear translation is not involved here.
Great. We have another question. What is the molecular function of PrEPL?
That is something we are looking into. We have a lot of thoughts. The first thing that will solve it is to look for physiological substrates, and that's why until now we know that it's a thioesterase, and we are currently on the verge of optimizing a technique called metabolic labeling, where we believe that PrEPL cleaves palmitoylating the palmitoylate groups of its substrates, and is basically doing its own the thioesterase activity. So that's something to be proved, and needs to be proved, and we are looking for the substrates.
Okay, great. Another question, did you assess the mitochondrial DNA levels altogether, given that there are only 13 proteins encoded by mitochondrial DNA in mammals, including only 2 subunits of the complex 4?
Sorry, I couldn't hear. Can you repeat it again?
Sorry, did you assess the mitochondrial DNA levels? Given that there's only a few genes encoded there?
No, we did not specifically look into mitochondrial DNA. We just checked the expression of the certain genes which are encoded by mitochondrial DNA, and they were found to be upregulated. And what we believe is that since the translation is reduced, and this also happens, that the increase in transcription of these genes can be the compensatory mechanism that is happening because of the impaired translation.
Great. Another question, are there any palmitoyl modifications to PCSK1 or other secretory pathway genes?
Yes, there are. So PrEPL is firstly known to be involved in regulatory secretory pathway, as I told before, and also it is known to be linked linked with AP1. Basically, this AP1 is important for making the secretory vesicles. And this AP1 is attached to the secretory vesicle via PEI4P, where we think that, and we know that when we knock out the PrEPL, the AP1 secretion or AP1 diffusion out of these secretory granules keeps on increasing. So that's something can be the possible substrate we thought initially, but yeah, there are PIP4 involved in secretory pathway and a lot of other proteins that are involved in secretory pathway, which are palmitoylated.
Okay. Great. Well, I don't see any additional questions. I'll just thank you for your time and for the presentation. And we'll move on to the final presentation of the symposium from Marnie Blewett, investigating the epigenetic regulator SMID-D1 as a potential therapeutic target for the treatment of PWS.
Thanks very much to the organisers for giving me this opportunity to present our work on gene activation-based therapy for Prader-Willi syndrome. So my name is Marnie Blewett and I work at WeHi. So what we're really trying to do is to try and switch genes back on again to treat Prader-Willi syndrome. We know that Prader-Willi syndrome patients are missing in some way, shape or form all of these genes on chromosome 15, but they also all have the imprinted maternal allele. So if we could switch these back on again, then this is a potential treatment. And we believe that this might be most beneficial during the early neonatal period when brain development is ongoing. although it may also be relevant later. So how could we possibly do this then? Well, presumably by removing the epigenetic marks that are silencing this cluster in the first place. So these epigenetic marks include DNA methylation, H3K9 trimethylation, ZNF274 is a factor that integrates with H3K9 trimethylation. And what I want to tell you about today is that this also includes SMUCHD1, a protein that my lab works on. So SMUCHD1 is shown here in the center. It's a large chromosomal ATPase, and this is relevant because, of course, an enzyme is going to be easier to drug, and there's a kind of a well-trodden path. So what we've mostly been working on for the past 10 years or more is SMUCHD1 in a mouse context, and in particular during early embryonic development. So when you have an animal that develops without SMUCHD1 during this critical period, then you activate all of SMUCHD1's target genes. You fail to repress them, and this includes the inactive X chromosome, the dosage which is inactivated for dosage compensation in mammals, all the HOX genes, which are important for segment identity and patterning, the clustered protocoherins, important in the neural system, and of course the Prader-Willi syndrome cluster, which is why I'm talking. So here, when you don't have SMUCHD1 during early embryonic development, you have activation just of the MAGEL2 end of the cluster, but do not affect the SNRPen or UBE3A end of the cluster. So for SMUCHD1 inhibition to be relevant to treatment for Prader-Willi syndrome, we need to know that these genes are important in disease. So here's the evidence that we have that these genes are important. So in particular, if you have a point mutation in MAGEL2 on the expressed paternal allele, this results in Prader-Willi-like syndrome, also now known as Schaff-Yang syndrome, described by Christian Schaff, which really phenocopies Prader-Willi syndrome with the exception of hyperphagia. If you have a point mutation similarly in McCoran3 on the paternal allele, it results in precocious puberty, which is found in some Prader-Willi patients. And in Nectin, the mouse model that is null for Nectin has a similar set of phenotypes to the MAGEL2 null model. And these, again, are very similar to Prader-Willi syndrome with the exception of hyperphagia. So we think that SMUCHD1 inhibition could target the majority of Prader-Willi syndrome patient symptoms, but we would need to treat in conjunction with treating the hyperphagia because this wouldn't affect the hyperphagia. So these are the questions I'd like to cover today. I'd like to first talk about whether or not it might be safe to target SMUCHD1 because I've just told you that it's got really important other targets around the genome, the HOX genes and the inactive X. I want to talk about whether or not it would be effective because we've predominantly worked on SMUCHD1 during early embryonic development. So what about if you remove SMUCHD1 later on, more relevant to treatment? And finally, what about humans? Is the same true in humans? So we took cells of the late gestation hypothalamus in the mouse and plated them down in culture to try and address the first two of these questions. And when we did this and deleted SMUCHD1 and we then harvested cells 5 days later after deletion and performed allele-specific RNA-seq so we can tell the maternal from the maternal allele. And what you'll see here, we're plotting, I'm plotting the maternal allele, so the imprinted one that's off. And when we delete SMUCHD1, we reactivate PEG12 and reactivate Nectin. MAGEL2 wasn't expressed, unfortunately, and we did not affect the SNRPen end of the cluster. And that's telling you that even when we delete SMUCHD1 after early embryonic development, because this is actually late gestation in a relevant cell type to Prader-Willi syndrome, we can still have reactivation. So it's likely effective. So we then looked at the rest of the genome here in an MA plot, and you can see that there really are very few differentially expressed genes. We don't have wholesale changes at all of SMUCHD1's targets. And this suggests maybe it might be safe. And so this is consistent with data that we've previously published in cortical neural stem cells, where again, we found the same thing. That is, we didn't affect the majority of SMUCHD1 targets, but we did affect the Prader-Willi cluster. So we then wanted to move to an in vivo model where we could have much longer term deletion, not just 5 days. So in this case, we used the hematopoietic system and we used VAV-CRE that deletes about mid-gestation, importantly after early embryonic development. And we aged the mice for up to 2 years and had 6 samples of female B cells to compare. And they're female so that we can make sure we would see changes on the X chromosome. And again, we see really limited changes. The blue are the differentially expressed genes. They include the Prader-Willi cluster genes and a few clustered protocoherins. MAGEL2 again was not expressed, but we don't see any upregulation of the HOX genes or the X chromosome. And this is really critically important because it suggests it might be safe to target SMUCHD1 after that early critical embryonic development time. So this shows us that if we take these two pieces of data together, that it might be effective to target SMICD1, because even when we delete after early embryonic development, we get reactivation of the Prader-Willi cluster genes. But most SMICD1 targets don't require SMICD1 continually to maintain their silencing, and this means that it might be safe. So next, we wanted to use a model that was again in vivo, but where we could finally measure MAGEL2, which is had been a little bit elusive for us. So here we used a MAGEL2 knock-in that also can be used as a reporter allele generated by Rachel Weverick. And so here it has a LAC-Z knock-in, and if you transmit it from the father, then the MAGEL2 expressing tissues are blue, and from the mother, it's off, and so you don't see blue when you stay with XGAL. We combined this with two different CREs, an inducible CRE, tamoxifen inducible CRE that we used to induce in the neonatal period, and secondly, a neural-specific CRE, which is not inducible, called Nestin CRE, much like VAV CRE. It's in the neural system here from mid-gestation, so after early embryonic development. And we then let the animals develop until after weaning and sectioned their brains and did XGAL stains. And so the negative control should have nothing when you maternally transmit. The positive control is the paternal transmission, lots of blue. And then the test is maternal transmission plus SMICD1 knockout, and asking what happens. So the first data here is the neonatal deletion with the tamoxifen inducible CRE. Positive control, negative control looking just as they should. And in the test animals here, you can see, it's very hard to see without zooming in, but zooming in, you can see in the thalamus here and the hypothalamus here, there's also some in the cortex. We see a limited amount of reactivation of MAGEL2 by these limited number of blue cells. So we wondered why we would have such a low reactivation. And when you go and check, it's because you have incomplete deletion in the brain, shown here by PCR. So really very incomplete, because we've still got at least 50% of the floxed allele present. So this is because of the blood-brain barrier. So even when you treat neonatally, the blood-brain barrier is established enough that you can't get tamoxifen easily into the brain. And if you have a high enough level of tamoxifen treatment to delete effectively in the brain, actually the animal dies because of tamoxifen toxicity, because even if that animal doesn't have the CRE-ER, it dies anyway. And so all we were able to get after trying 7 different treatment regimes were these 2 animals that survived with still very incomplete deletion. But all the same, we can say, even if we do delete post-birth, we can reactivate MAGEL2, although we can't really quantitate that here, and it's not a workable model. So we moved on to using Nestin CRE, which didn't require an induction, but was deleting after early embryonic development. Again, positive control, negative control. We have multiple negative controls. And then here I'm showing you 2 of our 5 test animals. And what you can see now is you can see that blue staining in the hypothalamus here and in the cortex and in these 2 animals. And if you zoom in, you can see it more clearly in the hypothalamus. So this was really nice data, and we've got 5 different test animals that show something similar. When we look at deletion, we can see complete deletion in the brain now, as we would hope. And importantly, we have survival of both males and females, suggesting that it's safe to be able to delete SMICD1 and may be safe to target SMICD1 after early embryonic development. So what I've shown you is that based on the gene expression studies and also based on the viability, it might be safe to target SMICD1 because we don't seem to reactivate its really major targets of the HOX genes and the inactive X chromosome. I've shown you also both in vivo and in vitro that it might be effective to target SMICD1 after early embryonic development. So the final part is it relevant to humans? So what we've done here is delete SMICD1 using CRISPR-Cas9, and I'm showing you this data in clonal 293 T cells. SMICD1 is in magenta, and it's gone by IF in the nucleus, as you'd expect. And so we then measured by qPCR, and what you can see is that in these replicate clonal cell lines, we induce MAGEL2 with SMICD1 knockout, Macorin 3 as well, but we don't change UBE3A expression, and nectin was not expressed here. And so this suggests that in human, SMICD1 behaves just as it does in mouse. That is, it's continually required to repress this one end of the Prader-Willi syndrome cluster, and we have similar data in Ren cells. So now, as a work in progress, we've been doing this in patient-derived cells. These are Schaff-Yang IPS cells that we've used in collaboration with Christian Schaff. And so we have here, we've deleted SMICD1, and we have a series of cell lines that have got a variable amount of deletion of SMICD1. And what I hope you can see is that as you have an increasing level of deletion, you have an increasing level of expression of nectin and MAGEL2, but we have no change in expression of UBE3A, just like we've seen in all of the other contexts, mouse and the other human cell lines. So the expression of these genes, nectin and MAGEL2, is really too low in these cells for us to confidently score whether or not this is now biallelic expression. And so we're performing the same experiments in Schaff-Yang and Prader-Willi syndrome patient neural lineage cells. So what I've shown you is that we think that it's potentially safe to target SMICD1 because it's not continually required to silence HOX genes in the inactive X. We think it might be effective based on in vivo and in vitro data because we can reactivate the Prader-Willi syndrome genes, and we think the same is true in humans. So what we're doing now is we're really focusing on our proof of principle studies, particularly in those patient-derived cells, looking at gene expression both at the Prader-Willi cluster and genome-wide. And then we're going to come back and focus on the phenotypes in the mouse model to see if we can actually rescue any of those phenotypes with the level of reactivation we observe. This is partnered with an active drug discovery program. We've just finished a high-throughput screen to find SMICD1 inhibitors of about 300,000 compounds, and we have over 2,000 hits that we're now working on. And finally, also, we're really interested to ask how SMICD1 actually silences the Prader-Willi syndrome cluster and how it integrates with the other epigenetic marks here, because we think critical to being able to work out how to best target that imprinted allele and switch it back on again by thinking about how these factors work together. So it just remains for me to thank all of the critical contributors, in particular Megan Aminatov, Tamara Beck and Sarah Kinkle, whose data I've shown you today, our wonderful collaborators Christian Schaff and Stormy Chamberlain, and also the National Drug Discovery Centre and all the researchers in my lab and other labs that work on that drug targeting side, plus of course our funders, in particular the Foundation for Prader-Willi Research and Prader-Willi Research Foundation Australia. So thank you very much for your attention, and I look forward to your questions that you can ask now.
Great. Thank you so much, Marnie. That's a really fascinating. I know it's been quite the tour de force over the last couple of years. It's great to see the project developing. So we'll wait for questions from the audience. But, I was curious if you could comment a little bit more about one of your future directions, which is, how does SMICD1 interact with some of the other factors that we know are impacting the epigenetics of the region? Do you have any ideas about that, or how you'll go about looking at that?
Yeah, so at the moment we haven't done anything on that side in the human context. We've done lots of studies in the mouse context, and so if we base our presumptions on what happens in the mouse, we expect that SMICD1 is likely downstream of both DNA methylation and also other histone modifications. At least in the mouse context, it seems to be downstream of polycomb marks, so H3K27 trimethylation, and based on some other people's reports, H3K9 methylation. And the H3K9 methylation is not work at the Prader-Willi cluster, it's elsewhere in the genome. So that's what we really want to test in the patient-derived lines. So in the human context, ask, is it downstream of these other epigenetic marks? And we've also shown that, again, in the mouse context, SMICD1 regulates chromatin architecture. So is SMICD1 setting up a chromatin architecture which is itself required for silencing? That's our current presumption and what we'd like to test, but we need to do that in the context of the human locus because it is a little different to the mouse locus, even though so far, in terms of gene expression, SMICD1 seems to behave the same in both cases. Yeah.
Okay, so you're not seeing like a change in DNA methylation when you see this activation by knocking out?
So you can do, sometimes it depends when you knock out SMICD1. So if you remove SMICD1 in the early embryonic period, then you do affect DNA methylation. But if you do it after that, you still get gene reactivation, but without a change in DNA methylation. So its effect is independent, doesn't require a change in DNA methylation, which is why we think it's downstream. Yeah.
Okay. Great, thank you. Question from the audience. Did you perform any ChIP-seq to know any change in the methylation pattern on chromosome 15?
So we haven't done that in human yet. We'd like to do that in human. We've only done that in mouse. And so in the mouse context, we find that we don't really see any changes in polycomb marks, and we don't see changes in DNA methylation, but we do see changes in gene expression and see changes in chromatin architecture. We haven't checked every histone mark, so there are many more histone marks we could check. We've mostly focused on polycomb, so SMUHD1 seems to be downstream of polycomb, to H3K27 trimethylation and H2A-K119 ubiquitin, but there are a whole host of other epigenetic marks, and particularly relevant K9 trimethylation would be really useful to look at in the Prader-Willi cluster, I think. Yep.
Great. A couple of more questions. Are you only considering small molecule chemical compounds for inhibiting SMUHD1 as a drug discovery pathway, or also siRNAs, ASOs, shRNAs? Any of those?
So at the moment, our drug discovery pipeline is completely focused on small molecules because SMUHD1 is an enzyme, it's an ATPase, so that's quite a well-trodden path. And certainly for drug discovery, that's where we're focused, although absolutely open-minded to alternatives like ASOs in the future. In our models, so in cells and in mice, we're using whatever is working. So we've got CRISPR-Cas9 and shRNAs. We're thinking about ASOs. So whatever is going to work, particularly in the neural lineage cells, because, of course, they're harder to target than nicely replicating other cell types where you've got lots of options. Yeah.
Great. And Rob Nichols asked, does SMUHD1 affect any other imprinted clusters or parts thereof in addition to the PWS region?
So this we've done in mouse as well. So most things we've done in mouse. So in mouse, yes, but almost exclusively, or at least exclusively in all of the experiments we've done to date. In the animal that's born or in the embryo proper, we're only affecting the SNRPN cluster. The other imprinted regions that we're affecting are only in the placenta or in the pre-implantation embryo, and they tend to be the non-canonical imprinted genes. And we have some small effects at parts of the KCNQ1 cluster. So they are broader, but we don't affect them in the actual animal that's born, only in the temporary tissues of the placenta. And as I said, they're mostly non-canonical, and those non-canonical imprints, for those of you that aren't aware of them, are mediated by polycomb marks, by H3K27 trimethylation and H2A-K119 ubiquitin, which is actually part of what started us down that pathway of considering its SMUHD1's role downstream of polycomb rather than strictly DNA methylation, because we have this really, the biggest effect, or numerically the largest effect, is that these non-canonical imprinted genes.
And, within the PWS cluster, so your effect is mainly on maggoslunectin, macrorin, but not on SNRPN. So what is your explanation for that?
Yeah, it's a good question. So it's a theory. I don't have really good data on that. When you look at where SMUHD1 binds in the mouse cluster, it has many binding sites down that end and actually kind of towards the middle, but not at the imprinting center itself. And then maybe one or two right down at the UBE3A end of things. So I think it's probably looped out. I think you've got a different domain for the long non-coding RNA and therefore the SNO RNAs that are contained. And so SMUHD1's not affecting that in isolation. What I think will be very interesting to ask is, well, what about if you have combinations? What about if you remove ZNF274, for example, or K9 trimethylation? Do you now sensitize? Is there a synergistic effect of then removing SMUHD1 on top of that? So do you have better reactivation, for example, when you remove both? Even though we don't normally, SMUHD1 by itself, when you remove it, doesn't affect that end of the cluster. But I suspect it is through chromatin architecture and different kind of three-dimensional arrangements that we don't yet completely understand. But of course, that we'll have to test in cells.
Yeah, it'd be really interesting to look at, the G9A inhibitors in combination or others. Yeah. Okay.
Yeah.
All right. I think that's all the questions we have from the audience. Thank you so much for getting up in the middle of the night, joining us. We do appreciate that. And so all that is left is to thank everybody, all of our speakers, all of our attendees, all of you who participated, It was a fantastic set of talks. Please, again, take a look at the posters, which are also really exciting. We'll be sending out a link next week for the presentations in case you missed any of them. That'll go to all of our registrants. And just a reminder again to mark your calendars for next year, September 29th and 30th in Chicago. We're so excited to be able to see you in person next year. What a thrill that will be. And finally, on behalf of our patient community, I just want to thank all of you who are working in this field. I mean, you are the superhero rock stars to our community. We appreciate all of the work that you put in, and we thank you and have a great weekend.